JP sir.
Yes, sir.
Sir, we can start, sir.
Okay, sir, we'll start.
Yes, sir. Yes, sir.
Yes, ma'am.
Sir, உங்களது echo வருது.
Okay, ma'am. I'm addressing it.
Okay.
Okay, ma'am, we'll start. Okay.
Okay, sir.
Father, shall we start further?
Yes, sir. Sure.
Thank you, Father. Thank you. The main purpose of human life is to get educated by good thoughts and to tell those to the world even in a better way, so that the younger generation learns from them and inspires others to become good. A very good morning and warm greetings to all of you on behalf of the PG and Research Department of Biochemistry, Sacred College, Autonomous, Tirupattur. We take this opportunity to welcome you all to the Science Academies virtual lecture workshop on recent advances in microbiology and biotechnology. Prayer is the exhaling the spirit of human and inhaling the spirit of God. We shall begin this workshop with a short prayer. Let us all close our eyes and maintain 1 minute silence as we recite this prayer. Let us pray the God Almighty to bless us with his fullest choices during this workshop. Oh Lord, please help us to carry out this workshop in a better way so that there will be a fruitful learning throughout this 2 days workshop. Bless all the participants who are taking part in this workshop and bless them with your abundant blessings, and may this workshop enlighten each and everyone in their academic world. Thank you. Now I invite Dr. A.C. Gomati, Head PG and Research Department of Biochemistry, to hear the welcome address.
A very pleasant morning to everyone gathered here. First of all, I thank God Almighty who has enabled us to organize this virtual funded workshop. Welcoming is the culture. It gives me a great pleasure to welcome the dignitaries to this inaugural function of 2 days Science Academies virtual funded lecture workshop
बायपास वर्कशॉप अटेंड कर रहा हूँ, मैम ले रही है.
virtual funded lecture workshop
साउथ
on
इंडियन
recent advances in
हम लोग हैं.
yes sir.
Dear participants, kindly mute your audio and video as the inaugural session gets started. Kindly cooperate with the organizers. Kindly mute your audio as well as video. Ma'am, you can proceed, ma'am.
Okay. 2 days Science Academies virtual funded lecture workshop on recent advances in microbiology and biotechnology. I welcome all the eminent speakers from different countries and states to share their knowledge and vast experience with the students community. We are proud to announce that this workshop is being sponsored by Indian Academy of Sciences Bangalore, Indian National Science Academy New Delhi, and the National Academy of Sciences Allahabad. On behalf of the Biochemistry Department and Management, I express my sincere thanks to the team members. The true essence of humankind is kindness. There are other qualities which come from education or knowledge, but it is essential. If one wishes to be a genuine human being and impart satisfying meaning to one's existence, to have a good heart. I take this opportunity to express my hearty welcome on behalf of the Biochemistry Department to the person who walks along with us, provides us continuous support, and encourages us to achieve this level is none other than our Honorable Rector and Secretary, Reverend Dr. John Alexander, SDB. Still, we remembered his motivation, even he is not available today. Welcome, dear Father. Genuine people can do great things with uncommon perfection because they are no longer concerned about their own interests and their own reputation, and therefore they no longer need to waste their efforts in defending them. Yeah, this is none other than Reverend Dr. Maria Anthony Raj, SDB. He motivates us to think differently. Still, we remembered as encouragement, even he is not also available today. Thank you, dear Father. A person who is very efficient, genial, genius, and silent worker. This person is none other than Reverend Dr. John Bohr, SDB, academic guide, controller of examination, State at College. I take this opportunity to welcome him to this inaugural session. Welcome, dear Father. Men do not really live for honors or for pay. Their happiness is not in the taking and holding, but in the doing, the striving, the building, and the serving. I take this opportunity to welcome Dr. DJ Bhakyaraj, NASI, Honorable Scientist and Chairman, Convener of the workshop and resource person, Center for Natural Biological Sciences and Community Development, Bangalore, who will deliver his speech on biodiversity and arbuscular mycorrhiza fungi for sustainable agriculture. Welcome you, sir. I extend my hearty welcome to our resource person, Dr. Dipshika Chakravarti, Professor, Tata Innovation Fellowship, ASTRA Chair Professorship, DAE SRC Fellow, Humboldt Fellow, Department of Microbiology and Cell Biology, Associate Faculty, Biosystem Science and Engineering, Indian Institute of Science, Bangalore, who will share her deep insights and perspectives on the bacterial cell wall structure and function and quorum sensing. Welcome you, ma'am. This is the ideal time to welcome our next resource person, Dr. T.K. Adya, Director, South Asia Nitrogen Center, New Delhi, Co-Director for Science Stream, UKRI, GCRF, South Asia Nitrogen Hub, Professor, School of Biotechnology, Kalinga Institute of Industrial Technology, Deemed University, Bhubaneswar, Odisha. Who will deliver a speech on soil microbiome, biodiversity under our feet, and plant microbiome looking at the future. Welcome you, sir. I take this opportunity to welcome our next resource person, Dr. Harini Kumar KM, Professor and Head, Department of Biotechnology, University of Agricultural Sciences, Bangalore, who will convey his knowledge through lecture on role of biotechnology in crop production and biofuel for a sustainable future and entrepreneurship development. Hearty welcome to you, sir. Next, I would like to welcome the coordinator of this lecture workshop, Dr. Nias Ahmed, Assistant Professor, PG and Research Department of Biochemistry, Saidat College, Autonomous, Tirupattur. Welcome you, sir. I extend my warm welcome to Dr. A. Jayaprakash, Assistant Professor, UG and PhD Head, PG and Research Department of Biochemistry, Saidat College, Autonomous, Tirupattur. Welcome you, sir. Teams means together everyone achieves more. Author Henry Ford says that coming together is a beginning, keeping together is progress, and working together is success. Teamwork is simply stated that it is less me and more we. Now I would like to welcome my dear colleagues and also the participants who come from other institutions and colleges. Welcome you all. Alone we can do so little, together we can do so much. My heartfelt welcome goes to my dear student friends. Optimum is the faith that leads to achievement. Nothing can be done without hope and confidence. With that note, again, a warm welcome to all of you and hope that this workshop will be fruitful. Thank you.
Thank you, ma'am, for your wonderful and elaborate welcome address. The leader is one who knows the way, goes the way, and shows the way, says John Maxwell. A great leader has the ability to inspire action through proper guidance and mentorship and use his knowledge to educate others along the way. We have an eminent personality in our midst, Reverend Dr. John Borg, SDB, our beloved controller of examination and academic guide for this inaugural session. With due respect and honor, I invite our beloved father to give his presidential address.
So good morning, dear friends. I hope I am audible.
Yes, father, you're
Yes,
audible.
Father.
Yes, thank you. Just 2 weeks before, I was invited for another program in the same department of microbiology, and it was indeed a pleasure that this department is not staying where it is, but progressing in leaps and bounds towards excellence. The motto for this academic year for Sacred College is to progress towards excellence in the path of synodality. Synodality means that together, as Professor Gomadi, Dr. Gomadi, the PG HOD has rightly put it in her welcome address, the department works together towards excellence. And one of the ways in which it has progressed is through their research and publication and their collaboration with eminent professional bodies in the context of workshops and seminars and conferences. I am indeed very happy to see that they are in collaboration with different institutes in organizing this online workshop with the topic recent advances in microbiology and biotechnology for 2 days. And I am also indeed very glad that they are being sponsored by Indian Academy of Sciences, Indian National Science Academy, and National Academy of Sciences. I would like to, first of all, acknowledge the great work put by this department under the headship of Dr. Gomadi and also Dr. Jayaprakash, the UG and PG HODs, respectively. And they have indeed worked very hard and make sure that this seminar sees the day. And a special word of appreciation to Dr. Nias Ahmed for coordinating from the side of Sacred College, and a great appreciation and a big thanks to Dr. DJ Bakiraj, the convener of this workshop and a very eminent scientist and chairman, okay, from Bangalore, from the Center of Natural Biological Resources and Community Development, for having consented to work with our team in Sacred Heart College to march towards disseminating knowledge about the recent advances in the fields of microbiology and biotechnology. I would like to acknowledge also the work rendered and the collaboration rendered by other faculty members of this department who are very much interested not only in teaching but also in research, consultancy and publications at a very quality level. Indeed, I am also glad that we have also other eminent speakers of the day. We have Dr. Deepshika Chakravarti, who will be giving us the first session on bacterial cell wall structure and function and quorum sensing, and followed by the coordinator himself, Dr. Bhakyaraj. And we also have Dr. T.K. Aditya, who will be speaking on soil microbiome biodiversity under our feet and on a role of biotechnology in crop production. We also have another speaker, Harini Kumar, who will be speaking to us tomorrow, the last session on the topic biofuel for the sustainable future and entrepreneurial development. It is indeed very fortunate and glad to see that various and diverse fields of biochemistry, biotechnology, and microbiology are being covered. And surely this will be a platform in which we can exchange knowledge, collaboration for better growth in academics, research, and various other avenues through which we can increase our own landing or standing in the academic field. So I'd like to congratulate and wish the Department of Microbiology as it marches towards such great initiatives. May God be with them. May God bless them. May they be always rewarded for all the ventures that they have. Surely this great culture of very professional academicship will be given also to the students of this department who are UG, PG and research students of this department. And this will in turn will emanate to other departments in the college.
Academic workshop
which will help the college to march towards greater heights. In this workshop, I would like to also announce to the crowd here that our Sacred College has been ranked 82nd in the NIRF ranking, the recently published NIRF ranking in the whole of India, and it is seen as one of the topmost arts and science colleges in which teaching, learning, research, consultancy, publications, and greater collaboration is happening. Though we belong to a very rural background, but we don't compromise on any of these standings in higher education. So thank you once again for inviting me for this inaugural session, and wish you all the best. And in a very special way, thank the HODs and resource persons. In a very special way to Dr. DJ Bhakyaraj for making this workshop possible, and also to Dr. Nia Sadaman. All the best and thank you.
Thank you, Father, for your warm and ornamental words. Certainly, your words have given us the encouragement to march towards excellence. Thank you all for your support to this department. Thank you, Father. It's my great pleasure to invite Dr. I. Niyaz Ahmed, the coordinator for this workshop, to provide an overview about the 2 days lecture workshop.
A very good morning to one and all. Really, it's highly fortunate that I get a chance to interact with the participant. Traditionally, this overview about the workshop has to be delivered by our convenient Dr. Joseph Bhakyaraj sir. This emergency is unable to join with us. As per his expertise and as per his instruction, I would like to share some of the
Printing வந்துட்டே இருக்கு chat.
Science Academics Workshop. Indian Academy of Sciences was founded in the year of 1934 by Sir C.V. Raman, the Nobel laureate. Its objective include promoting the progress and upholding the cause of science with adolescent separate branches. Starting in the mid of 90s, Indian Academy of Science had initiated several activities for the benefit of students and teachers of science all over the country. These include summer research fellowship for students and teachers to work with the fellows of the academy and other faculty in the country. Refresher course for 2 weeks duration for teachers. Lecture workshops around 2 to 3 days duration for the students as well as the teachers. The Indian National Academy, the Indian National Science Academy, New Delhi, and the National Academy of Science, Allahabad, joined with this Indian Academy of Science, Bangalore, for conducting 3 of the programs, that is summer research fellowship, refresher course, and a lecture workshop. With the short note, and we are highly fortunate to hold the Science Academies sponsored lecture workshop for the consecutive 3 years.
இந்த e-tools வந்து நான்
lecture workshop
அதை நீங்க
followed by
கண்டிப்பா அப்ளை பண்ணுங்க.
last year and this year we are conducting
500 ரூபாய் கேட்பாங்கன்னு
virtual
நினைக்கிறேன்.
mode.
அது வந்து
This
நீங்க
kind of
அட்டெண்ட்
lecture
பண்ணீங்கன்னா
workshop and refresher course
அதாவது
can be
ஏன்னா
conducted
நம்ம
by
ஏன் இதை
those
நான் சொல்றோம்
institutions
அப்படின்னா
like
ஹை
us.
இம்பாக்ட் ஃபேக்டர்ல
Help
நம்ம
of
பிளான் பண்ணிட்டு இருக்கோம் ஜேர்னல்
fellow
போடுறதுக்கு.
members
Sir,
of
may I,
அதுக்கான
Indian Academy of
டயக்ராமேட்டிக்
Science,
ரெப்ரசன்டேஷன்
National
சார்ட்
Academy,
இது
Science
எல்லாமே
Academy,
வந்து யூஸ்
the
பண்றதுக்கு
National
என்ன மாதிரி
Academy of
சாஃப்ட்வேர்
Sciences,
யூஸ் பண்றாங்க,
you can go through
எப்படி
the
அதை வந்து
details of the
ப்ளே
fellows
பண்றது எல்லாமே அவங்க கொடுக்குறாங்க.
via their
நான்
respective
இன்கேஸ்
websites
அந்த
of each
ப்ரோக்ராம் ரொம்ப
academy.
டிலே ஆயிட்டு
They are
இருந்ததுன்னா நான் என்ன
senior
பிளான் பண்றேன்னா அந்த
scientists
ப்ரொஃபசர் செந்தாமரை கண்ணன்
interested
அண்ட்
to
டாக்டர்
collaborate
இந்திரன்
for the betterment
2
of
பேருடைய மெயில் ஐடி நான் வாங்கி தரேன்.
in this
நம்ம வந்து
juncture. I would
பர்சனலாவே
like to
மெயில்
thank
அனுப்ப
kindly
சொல்றேன்.
mute your mic,
Mentor
sir.
Dr.
சப்போஸ் வந்து
Joseph Bhagiraj sir
ஒரு இம்பாக்ட்
for
ஃபேக்டர்
providing
ஜேர்னல்ல
me this
பப்ளிஷ் பண்றதுக்கு
opportunity to
என்னுடைய
conduct
பேப்பர்னுடைய
this
ரைட்டப்
kind of
வந்து எந்த
lecture
அளவுக்கு ஹெல்த்தியா
workshop
இருக்கணும்.
frequently in our
Hello.
institution.
ஈவன்
I would like
ரெஃபரன்ஸுக்குலாம்
to
கூட
thank
சாஃப்ட்வேர்
Dr.
இருக்கு.
Deepika
Yes,
ma'am
sir.
நீங்க அந்த
for
ரெஃபரன்ஸ்
her
சாஃப்ட்வேர் வந்து
availability
என்ன பண்ணலாம்னா
in the
இயர்
lecture
வைஸ்ல
workshop
ஃபில்டர் பண்றது, ஆத்தர்
schedule
வைஸ் ஃபில்டர் பண்றது,
schedule in the
ரெண்டாவது
tomorrow.
இயர் வைஸ்
Can all
ஜேர்னல்
mute the
வால்யூம்
mic?
வைஸ்
I'm
எல்லாமே
sorry.
From Bangalore to
Dear
Chennai.
What was the
பேசினாரு.
part?
நான் அது
Dr.
Thank
Niyaz
each and
Ahmed at
everyone
all.
who
This
are
is
நான் ஈவினிங்ல
Kanya.
தான் அட்டன் பண்ணிட்டு
associated
இருக்கிறேன்.
with this
ஈவினிங் 6 to 7 தான்
lecture
நடத்திட்டு
workshop.
இருக்கிறேன்.
We have 4 different eminent
வேணா என்னுடைய
speakers with us
மெயில் ஐடியில வேணா நீங்க
during
போய்
this 2 days
பாக்குறீங்களா?
lecture
நீங்க
workshop.
வேணா
Yes, sir.
ஈவினிங் அட்டன் பண்றீங்களா?
Yes, sir.
இன்னைக்கு நடக்க
Of
13ஆம் தேதி
Dr.
வரையும்
DJ
நடக்குதுங்க.
Bhagiraj
அது
sir,
6 to
one
7
of
தான் ப்ரோக்ராம்.
the academy fellow,
Yes,
honorable
sir.
fellow, followed by Jipsha ma'am. Also, she is also a fellow of the National Academy of Science, followed by Adya sir, also a member, a
மெயில்
fellow
ஐடியில
of the
போனீங்கன்னா
academy.
MS Teams
Followed by Dr. Arini Kumar sir from the University of Agriculture Center. They are
ஆப்
going to
தாங்க
enlighten us in
மைக்ரோசாஃப்ட்
the 2 days
டீம்ஸ் சொல்லிட்டு.
lecture workshop with their
Sir, Mr. Arun
expertise.
Gandhi.
இல்ல பட் இப்போ என்னன்னா
Mr.
நான் ஆல்ரெடி
Arun
மொபைல்
Gandhi.
இன்ஸ்டால்
To
பண்ணிருக்கேன்.
thank each and
மெயில்
everyone
இருக்குது.
who are associated
ஆனா
with
மெயில்
the
ஐடியில
Arun
லிங்க்
Gandhi,
ஓபன்
sir.
பண்ணனும் எங்க வேணாலும் ஓபன் பண்ணிக்கலாம்ல?
execution of this
Mr.
particular
உங்க
Arun
சிஸ்டத்துல
Gandhi.
இல்ல என்னுடைய மெயில் ஐடியில ஓபன் பண்ணுங்க. உங்க செல்லுல பாருங்க.
Yes, sir.
மெயில் ஐடியில
Dr.
ஓபன் பண்ணி
Gomti,
அந்த மெயில்
Yes,
லிங்க்
can as
கொடுத்திருக்காங்க
a
எனக்கு.
host you can
அந்த லிங்க்
mute.
நீங்க
You can
உங்க
mute
மொபைல்ல
him.
கிளிக்
Ma'am,
பண்ணுங்க. ஜஸ்ட்
actually,
உள்ள போதான்
ma'am,
மட்டும்
actually,
பண்ணிக்கலாம்.
Nia sir is the
Yes,
host.
sir.
Yes, sir.
Yes, sir.
Dr. Niyaz, kindly mute. Yeah, I couldn't hear you at all.
ஆஹா, அப்படியா?
I only heard the background speech.
ஓ லேப்ல லேப் பார்க்கலாம்ல நீங்க? அப்ப லேப்ல நீங்க என்ன பண்ணலாம்னா
Yes,
டைரக்டா
sir.
என்னோட மெயில் ஐடிக்கு போயிட்டீங்கன்னா அதுல
Yes,
நீங்க
sir.
மைக்ரோசாஃப்ட் டீம்ஸ் உங்க
Kindly
லேப்டாப்ல
mute
இருக்குதா?
the other participants. Yes,
அதுல
sir.
போட்டு இன்ஸ்டால் பண்ணி லிங்கை ஓபன் பண்ணி பாருங்க.
Yes, ma'am.
ஓபன்
Kindly
பண்ணீங்கன்னா அதுல
remove
தெரியும்.
him.
நேத்து
Dr.
வந்து
Kindly
டாக்டர்
mute
இந்திரன்
the
கொடுத்தாரு.
participants,
இன்னைக்கு வந்து
sir.
ப்ரொஃபசர்
Mute.
செந்தாமரை கண்ணன்.
Kindly
சாயந்தரம் 6 to 7ங்க.
mute Dr. Arun Gandhi.
புரியுதுங்களா?
I couldn't
Arun
hear
ஏன்னா
Arun
you.
நேத்து
Gandhi.
வந்து
Yes.
மைக்ரோசாஃப்ட் விசோ அப்படிங்கிற சாஃப்ட்வேர்ல போன் பண்ணி நேத்து பேசினாருங்க.
Sir,
அது
Mr.
எதுக்காக
Arun
அது
Gandhi.
Yeah, ma'am.
Yeah, kindly unmute.
Sir,
Unmute
Nia sir,
yourself and
உங்களோட
mute
off
Arun
of
Gandhi,
the
sir.
vote of thanks audible-ஆவே இல்ல.
This is fine. This is fine.
Ma'am,
Yes.
Deepika ma'am, can you
Hear Dr. Niyaz
want to
Ahmed.
hear it again, ma'am? We shall ask Niyaz
No,
sir
no,
to
no, it's
elaborate.
okay. It's okay. No
Okay.
problem.
Shall we proceed?
Yeah, no issues at all. Yeah.
Okay, ma'am. Thank you, sir, for your brief elaboration. I thank our beloved father, Reverend Dr. John Bohr, for his presidential address, Dr. A.C. Gomati for her welcome address, and Dr. A. Niyaz Ahmed for the brief overview about the workshop. I request all the participants to mute your audio and video as we proceed to technical session of the workshop. Now I hand over the session to Dr. A. Pungode for technical session 1. Thank you all and have a fruitful day ahead. Dr. A. Pungode?
Thank you. Yes, sir. Yes, here
Yes, ma'am.
only,
Yes,
sir.
yes, ma'am. Thank you, ma'am. Thank you.
So,
Kindly take over the session.
yes, sir. Dear dignitaries, professors, participants, and beloved students, Once again, a very pleasant good morning to one and all present here. So all the participants for today programs, workshops on recent advance in microbiology and biotechnology. Now it is my great privilege and feel happy to introduce the resource person of the first technical section, Dr. Deepika Chakravarti. She did a triple major BSc degree in chemistry, zoology, microbiology at the LAD College, Nagpur University in the year 1989. She did her MSc degree in microbiology with merit at LAD College, Nagpur University in the year 1991. She did her PhD degree in biotechnology at the National Center for Cell Science, Pune University in the year 1999. She is currently working as professor, Department of Microbiology and Biology, Indian Institute of Science, Bangalore. She has several research positions like Junior Research Fellow at the National Center for Cell Science, Pune in the year 1994, Senior Research Fellow, Visiting Scientist at the Department of Microbiology and Immunology at Medical University, Japan in the year 2001, Hamfold Fellow at the Institute for Clinical Microbiology, Immunology and Hygienic, Friedrich Alexander University in the year 2004. She has got several awards like Yoga Maya Devi Award for Research in Medical Science for outstanding work by Maharashtra Cultivation for the Associations of Science, Tata Innovation Fellowship, Professor S.K. Shorterji Award in the year 2020, Fellow of Indian Academy of Science, Bangalore, Astrashar Professorship Award, National Bioscience Award. Uh, she has presented several papers in various workshops, seminars, and conferences. She has published many papers in various national and international reputed journals. She also one of the editor or editorial board member of the various journals like Innate Immunity, Sage Publication, Resources, etc. She have six patents in her credit in research, especially for developing vaccine against Salmonella, a novel PCR diagnosis for Salmonella typhi, and a novel mechanism of drug delivery to intracellular pathogens. She has several committee members and also the scientific board of studies from outside of IIC, like Board of Study Christ University, DB nominee, DST, and WSO sub committee members, and also the grant review for international granted. She has several professional membership like Molecular Immunology Form, International Society of Endotoxin Research, American Society for Microbiology, Indian Associations of Cell Biology, and Indian Associations of Basic Medical Science. So we are very much proud to invite such a wonderful personality as a resource person for this session. Welcome you, ma'am. Dear ma'am, once again, give warm welcome for here to present a valuable lecture. Thank you, ma'am.
Thank you, Dr. Pungotai, for such an elaborate introduction. I hope that you can all hear me.
Ma'am.
Yes,
Yes,
ma'am,
ma'am.
audible.
Thank you so much. So, Dr. Pungotai, I have how much time? Sorry, how much time do I have?
1 hour, ma'am. 45 minutes to 1 hour, ma'am.
Sure. So now it is 10. So by 11:05, right?
Yes, ma'am.
Okay. Now what I'll do is, first of all, I'll enable me to do the screen sharing. Dr. Niaz, you have to give me permission.
Yes, ma'am. Yes, ma'am. Please proceed, ma'am.
Okay. Post disabled participant screen sharing, it is showing.
One second, ma'am. I will check.
Host disabled participant screen sharing. That is what it is showing. Kindly enable it.
One second, ma'am.
Sir, kindly mute all other participants except the speaker. We are facing a lot of difficulties. Ms. Samna KP, kindly mute your mic.
145.
Ma'am, is it unable to share? Am I right?
It is showing host disabled the participant screen sharing. 145.
Who's Samna?
You have to enable it.
Allow parties to.
Click allow participants to share the screen, sir. Allow.
It is still disabled.
Oh, 1 second, ma'am. 1 second, ma'am. Ma'am, now you can check, ma'am.
Yeah, sir. Kindly mute all other participants, sir, except the speaker and the moderator.
Yes, sir. Yes, sir. Ma'am, now you can able to share it, ma'am.
I can't share the screen, sorry.
Oh, wait a second. Meetings.
And I can't share the screen.
Oh, 1 second,
No.
ma'am. I will address it. I'm checking it down. General. Share screen, share all. Share screen, enable all participant. कहाँ है इसमें? JP sir.
Yes, sir.
அதில்
Any issues?
நீங்க ஏதாவது ஷேர் பண்ணி ட்ரை பண்ணுங்க சார்.
1 second. Enable பண்ணிட்டீங்களா? You
enable
have
பண்ணி தான்
enabled all
இருக்கு
the
அதுல.
participants. Ma'am, ask ma'am to try again.
இல்ல நீங்க
Yeah, I'm trying. No, it is
Ma'am is
showing
trying.
host disabled participant
Oh, okay.
screen sharing.
Again it is showing that. Yes, sir.
லேபர் போட்டோகிராஃபில என்ன பிளான் செய்றீங்க? நாங்க வந்து
Yes, sir. It shows only host can share this meeting.
stranger class வச்சோம்.
எங்க போய் சேஞ்ச் பண்ணணும்னு தெரியலையே. எல்லாமே எனேபிள் தான் காட்டுதுல.
Otherwise, ask ma'am to share the PPT and you can.
Scale, share all windows, automatically share.
I can't share the slides because it's too bulky.
Okay, 1 second, ma'am. 1 second, ma'am.
Yeah.
I think we have to do some minor adjustment in settings, ma'am. Then it will work.
Yeah.
Ma'am, now you can try, ma'am. Dipshika,
Not
ma'am.
trying. 1 minute. Oh, great. Done.
Okay, fine, ma'am. Fine. Proceed, ma'am. Please proceed.
Thank you. Okay, colleagues, thank you very much. So first of all, let me extend my deep appreciation for the Sacred Heart College, Dr. Gomati, Dr. Niyaz Ahmad, Dr. Jayaraj, Jayaprakash, all of you, and plus Professor Bhagyaraj, who is my great friend. And we had done so many conferences and these kind of academic lectures together. But, this is my first time to be there in the Sacred Heart College. A long time back, I got an invitation from, again, one of my very good friend, Dr. Brito Das, and somehow due to some emergency, I couldn't visit your college. So this time also, I think this is online, but hopefully we will get sometimes, to meet all of you and all your students, colleagues. So I wish Sacred Heart College the very best wishes, and Reverend Father, thank you very much for this invitation. Now let us, I am a microbiologist, medical microbiologist or infectious disease research by training. And what I understood about the life of the bacteria, it is something like this. There is absolutely no way that we can say that these so-called unicellular prokaryotes are any way less intelligent than the so-called higher mammals. Their ability to, in fact, create such a big havoc and also play a very important life in making us what we are today due to our flora inside the gut is all because of their community behavior. So I'm going to throw some light on the community behavior, that is a quorum sensing. And what we will do is, I see that there are so many participants, 141, which is very good. So we will have a lot of discussion session. That is what I love the most. In between asking questions would have been preferred if I was in person, but now because of this, online, we will take the questions at the end, and I would request all of you to then unmute yourself and tell your name and pose the question. You can also pose it in the chat box, but let us have a very wonderful discussion sessions, because that is what I expect from these academic workshops. It should ignite the questions in all of you, and it should lead you to do really good work. In case you're not able to hear me, then I would request Dr. Niyaz Ahmad or Dr. Gomti to simply let me know and alert me so that I can go ahead. Okay, so what do you understand by this word quorum sensing? Quorum sensing, so let me put my pointer option. Okay, so the word quorum, it means that you are having a community. That is what is meant by the word quorum. What is quorum sensing? So what we are going to understand here are three very important questions. First, what is quorum sensing? Why one should get interested in quorum sensing? and what do we know about its history? Because I'm sure that your microbiology and cell biology department or your microbiology, you start teaching the students with the history of microbiology, which is one of the most important part for them to give the gratitude to all the microbe hunters because of whom we are learning microbiology today, are doing research on microbiology today. So what is meant by quorum sensing? Theoretically, you know that a bacterium divides into two. So 2 bacteria divides into 4. So there is a logarithmic progression. So you give them nutrients and they grow. And then what you get is something known as a stationary growth phase. The question always came that these nutrients, are these the only one which are going to have the only one which are going to get you the stationary phase? And the answer is no. Why the answer is no? Because it is just not the nutrient which determines that the bacteria should be growing in the stationary phase. The most important is it is about the signals. Now, what do you understand by the signals? Why a bacteria cannot grow, grow on and be infinite in their growth? Why there has to be a so-called stationary phase? And what is the use of the stationary phase? And what are the signals that we are talking about? Now, the signals are nothing but the signal which the bacteria secrete to tell the other group of the bacteria that we are now enough in number and now we should stop growing. So the growth, whatever is written in the textbook, it is because of the depletion of the nutrient and accumulation of the toxic waste product is absolutely not correct now. And we know that how important is this bacterial community to do the function that it is doing. So the signaling, the signal that it secretes goes like that. A bacteria is going to make this signaling molecule, and these molecules are known as a quorum sensing molecule. Now, the word quorum sensing has become, it's a very hot topic now. It means that a quorum is complete, So this word had been used so much in the parliament to say that now we can start the proceeding because the quorum is complete. So quorum means the defined number. It is all about the number game. How many number is good enough to start the meeting? How the number, the apt number. So it is not about more or less. It is not that 100 can be a quorum and 10 is not. If 10 is the number for that given meeting, then 10 is the quorum. If 100 is the number for that given meeting, then 100 is the quorum. So it is about the number that is required to do the function and effective way of delivery. So that is the quorum. So how bacteria attends? We can communicate with each other, talk to each other and get the things done. But how do bacteria communicate? Now, this communication with the bacteria is so important that if you can stop this communication, you can tackle anything that you want. And it is only because of the communication, these bacteria, they achieve what they want. How do they do this communication?
Ma'am, sorry for
Hello,
disturbance.
is there a question?
Ma'am,
Yeah.
sorry for
Is there a question?
No, ma'am, your slide not moving, ma'am. Only the first slide is only visible.
Oh, is it?
Yes, ma'am, yes.
Oh my God. Okay, then in that case, what I have to do? Can you see this?
Now only clear, ma'am. Now only visible one.
Now it is visible, na? So
Yes,
when I'm
yes.
putting it in the full slide mode, it is not visible. Okay,
Okay,
then we
now-
have to go this way only.
Okay.
Now
ma'am,
can you
fine.
see? You can see that, right?
Yes, yes, ma'am.
Okay,
Now
great.
see.
Then we will move not in the full screen because I think it is the bandwidth. So we will go by this, this mode. Okay, thank you.
Okay.
Thank you for telling. Yeah. Right. So now, this is what I was talking to you, that the bacteria will be going in this logarithmic phase from 1 bacterium to 2, 2 to 4, 4 to 8. But then where do it stop? Why do you get this so-called, the stationary phase? It is only because of the signals that it produces. Now, what is written in the textbook is that the nutrient is depleted, toxic metabolites are produced. That is not the case. It is because now the bacterial numbers are enough to start the processes of what it is destined to be. And the signals are known as a quorum sensing signals. This is a language for the bacteria to cross-talk with each other. Okay. Now, what are these signaling molecules? That is very interesting. And how these molecules becomes a language for the bacteria. Now, bacteria secretes this molecule. We will understand that procedure by which they secrete it. They also have a mechanism to export these signaling molecules, and they are exporting this signaling molecule so that other bacteria in the group can sense it and signal it. See, remember, if you have to communicate with everybody, you need to speak out. That means something has to be sent out. The message has to be sent out. Now, the bacteria, how do it send out its message? It sends out the message with the help of these so-called quorum sensing molecule. And they make the molecule, they send it out so that the entire community can benefit. Now, the biggest lesson from the bacteria that mankind should learn is the way by which they can have a very good communal life, community life. That means everybody helping each other. And that is the reason why it is extremely difficult to get rid of the bacteria, be it a pathogenic bacteria or be it anywhere. What is the reason that we are not getting? What is the reason that we are not getting rid of the infectious disease and you are getting troubled by the antimicrobial resistance, all the MDRs and the XDRs? It is only because that their communication is so strong that whatever weapons have been designed against them, they're going to fight it together and emerge as a superbugs. And there is no doubt about it. A right to live is also for these bugs. Okay. So we have to think now very smart to understand how do you stop this war between the bacteria and the human. Now, these signaling molecules are something which is an amazing set of resources that one can use. Now, the bacteria, once it reaches a certain density, now what they do is they start making this molecule and the other bacteria senses it. Now, how do they know that the number is correct now and now they should stop growing? Because the extracellular concentration of this particular quorum sensing molecule is equivalent to the intracellular concentration. So once that sensing system achieves an equilibrium, the bacteria comes to a halt in terms of its proliferation. And then they will stop the growth. And when they stop the growth, they start doing their differentiated function. Now, what is the differentiated function that I'm talking about? It can be anything like production of a color, to production of antibiotics, to production of an enzyme, to production of, say, light. Anything it can be. The product that is a good will be produced when the bacteria are in the adequate number. That means that they have achieved the quorum. Now, you know, this word quorum was introduced much late, but the processes were well known from the day the earth was, the first life on the earth was formed some 10 billion years ago, and then the bacteria that was the first one to come to the life, which populated the earth, and from then on it was there, the community behavior was there, but the only thing is that we understood it very late. So what do we know about its history? Now, the history of the quorum sensing, kindly mute, sorry, because it's for the benefit of all the others who are listening. Now, the history is extremely interesting. How it is interesting? Many of you as a child might have read 20,000 Leagues Under the Sea. It's a very beautiful book, story book, where it was mentioned about the tale of Milky Seas. You know, the tale of Milky Seas is not a fiction. It is a fact. Whenever these, now also when the NASA takes a picture from their satellites, what you see is a 250 kilometer stretch of the blue illuminating patch near the Indian Ocean. Now this blue illuminating patch is always there. It never drifts and it always illuminates. Now, what are these blue illuminating patch? In the same illuminating patch, all of you are in Chennai and might have been visiting the Marina Beach. It was noticed that in the night, when the waves come, there are illuminating waves. Now, these are nothing but the bacteria, which are called as a bioluminescence bacteria, illuminating bacteria, which glows. How this glow happen? How is the production of the light happens? Now, this production of the light is a product of the quorum sensing. That means when the bacteria achieves a certain density, they produce this light, which is now visible. Now, this particular bacteria, which opened up the research in the field of quorum sensing, is known as a Vibrio harvey. Now, Vibrio harvey is a species of Vibrio, like Vibrio cholera, but harvey is a free-floating non-pathogenic bacteria. This bacteria, Vibrio harvey, they come together and they lay down their matrix. Now, the marine microbiologists, they have sampled the water from this seawater, and they realize that the Vibrio harveys, they do not need anything else to survive, but there are a lot of zooplanktons and other kind of organisms from the sea, which actually lay down the matrix. They benefit somehow from this light production because a lot of food gets attracted towards the light to feed that small crustaceans or the other protozoans, etc. So this blue patch is the hallmark of, and it dates back to the history about the tale of the Milky Seas, which in this book was very beautifully 20,000 Leagues Under the Sea. If you get a chance, students, my young colleagues, kindly read this book. It's an extremely interesting book. How do you define then quorum sensing? In a very general term, it is nothing but a way for bacteria to count their numbers. Remember, colleagues, whenever you're doing even the experiments in your lab, or you're doing a biofermentation experiments, you're always heartbroken that the product is not formed, the transformation does not happen, or for the simple as the bacteria does not grow in the test tube, because we do not understand that how important is the number for the bacteria. So in a way, it is a regulation of the gene expression in response to fluctuations in the population density. It is a way for the bacteria to count their numbers, that the numbers are enough now, and now they can start their function. That is how it is defined. Or this communication between the bacterial cells, the language of the bacterial cells, is extremely important for the bacteria to achieve that quorum. And in late 1970s, the communication of the bacteria was understood. But actually, in 1920s onwards, they understood that bacteria are not as simple unicellular prokaryotes that we think of. Even hypothetically or mathematically, you will say that one bacterium can divide into 2 and 2 into 4. But you do this experiment where you dilute the bacteria, do a serial dilution so that your last test tube ends up in a 1 bacterium, and you try to initiate a growth curve from that 1 bacterium, you will not be successful. Why? Because even to initiate that growth curve, you need that minimum number or that quorum of a bacteria. So the communication between bacteria was understood, but it was quite difficult for anybody to, have a mindset that these simple unicellular, so-called unicellular bacteria can have such a complicated and sophisticated lifestyle, which even we, so-called the super brain human beings cannot even think of. So in these two systems, that is Vibrio harvey, which is a free-floating or the free organism, and Vibrio fishery. This Vibrio fishery is in association with deep sea fishes, deep sea fishes, squids, bobtail squids. They are associated with them, and these act as a light organ for these deep sea fishes, where in the depth of the sea, there is no light, there is no sunlight which can reach at the bottom of these deep sea beds. And if it is so dark, there will be no food available for them. So these kind of light organs acts as a food attractant. And this relationship with fish or the squid is a symbiotic relationship, because Vibrio, hello, kindly mute. Vibrio fishery cannot live alone, and the fish cannot live without Vibrio fishery. So these experiments are a bit difficult to do because these are deep sea experiments, and one has to really create those huge fish tank to mimic the deep sea. But the scientists have done that. And when they have gotten rid of this bacteria by antibiotic treatment, they realized that these fish cannot survive. They die because they cannot hunt for the food. So these light organs have become one of the very interesting way by which the deep sea fishes can hunt for the food, and the bacteria in turn, they get the nutrition from the fish. Now, what came to the rescue to the researchers to study on the quorum sensing is Vibrio harvey. Why? Because Vibrio harvey is free floating, and Vibrio harvey can be grown in the lab. You can grow the Vibrio harvey with the help of seawater medium. So these marine microbiologists were very smart. What they did was, when they led a cruise and they were sampling, they sampled this particular this blue stretch, and along with that they got some seawater. They filtered the seawater, mixed with some agar, and they made a seawater agar medium. Nowadays you have high media who gives the seawater medium. You can buy it from high media and you can grow this Vibrio harvey. And when they reach a quorum, they give this blue light. You can see it under the UV, or if you have a bioluminometer, you can see this blue light production. So the production of this blue light is an indication that the bacteria have reached the quorum. That means that the crosstalk is very successful between the bacteria. Now, let us look into the history of the quorum sensing. Tomatz in 1965, he first named this as a competence factor because the gram-positive bacteria, Streptococcus pneumoniae, that is a naturally competent cell. Now, Streptococcus pneumoniae is very famous. If I would have been in my physical class, I would have asked my young colleagues to tell you that why Streptococcus pneumoniae is very famous. Because Dr. Griffiths, Professor Griffith, he used this Streptococcus pneumoniae to prove that the DNA is a hereditary material. Okay, so but it is naturally competent. I mean, unlike E. coli, you don't have to do so many steps to make the cells, naturally competent and make your genetically modified E. coli. So you give them some DNA, they take it up. So Tomatz named it as a competence factor. That is, they're naturally very competent to take up an exogenous DNA. Then 5 years down the line, Nielsen in 1970, he used this bacterium Vibrio fischeri and termed this the molecule as an autoinducer. The production of the luminescence in the gram-negative bacterium Vibrio fischeri is controlled by the self-produced chemical signal, and that chemical signal is the one which he termed it as an autoinducer. He realized that definitely it is not contained within each bacteria. Bacteria has to secrete it out so that the community can get benefited. He termed it as an auto-inducer. That means it is induced by the bacterium and also used by the same group of bacterium. So auto-inducer. Then few years down the line, Eberhard in 1981, he identified the Vibrio fischeri autoinducer signal to be N3-oxohexanoyl L-homoserine lactone. Now this molecule, N3-oxohexanoyl L-homoserine lactone, or AHL, this homoserine lactone, is the first identified quorum sensing molecule produced by the bacteria. And Engelbrecht, 2 years down the line in 1983, cloned the genes which is responsible for the production of this particular quorum sensing molecule, that is a homoserine lactone, and he named it as a lux gene. Lux is the intensity unit for the intensity of light, and he termed these genes as a lux genes. He also cloned the gene for the signal which is generating this enzyme. He understood the receptor which can make this blue light production, and he termed it as a lux gene. Okay, so now how to go ahead from there? Who introduced the term quorum sensing? Just not very, few decades back, Fukua in 1994, only in 19th century, 1994, introduced the term quorum sensing to describe the cell-cell signaling in bacteria. And from then on, this word quorum sensing had been used left, right, center, and it has become not only a very important, area of research, but every single word that you do in microbiology, at the end of this talk, you will be convinced that if you are not understanding the quorum, that you cannot take a step forward in the area of research in microbiology. So in the Vibrio fischeri Luxi-Luxar system, it became a paradigm for the bacterial cell communication. In early 1990s, the homologue of the Luxi was discovered in the different bacterial species. So what we will understand in a very simplistic way, because I think that my esteemed teachers who are from different colleges, and my colleagues who are from maybe from the undergraduates and the postgraduate, it is good to go a bit slow, because you just try to now understand that how these molecules, and appreciate the fact that the bacteria can talk to each other. And you also appreciate the fact if you stop this crosstalk between the bacteria, then all the functions of the bacteria are compromised. They will not function the way they are supposed to be. Now, this is the chemical structure of the molecule. I have a colleagues who are also majoring in chemistry or have taken chemistry. It is one of the very beautiful structure, very simple. This is this lactone ring. It is an acylated homoserine lactone, which is produced by the diverse group of the gram-negative bacteria. And what you see here is this R side chain. Now, this R side chain is the acyl chain, which differs from bacteria to bacteria. This particular core structure or the lactone ring is conserved in all the different gram-negative bacteria. To give you this example, now this AHL molecule or acylated homoserine lactone, it is made by the bacteria. The bacteria exports it or secretes it to the environment. This is also taken up by the another group of the bacteria in a given consortium or in a given solution or a mixture. Now, let us understand that what are the different kinds of the acylated side chain. So, for example, in the Vibrio fishery Luxi system, you have, this is the acyl side chain. Yeah? So what you appreciate is this lactone ring is conserved, okay, everywhere. But the acyl side chain is something which is differing. So Vibrio fishery acyl side chain is different from Pseudomonas aeruginosa acyl side chain. Now, Pseudomonas, we will study, very soon, that Pseudomonas aeruginosa is a gram-negative bacterium. You might have heard of this bacterium. Now, this has a hierarchical quorum sensing system. That means there are two quorum sensing systems. If only the first works, the second quorum sensing system kicks off. You cannot have the first one inactive and the second one working. So that is why it is a very beautiful hierarchical quorum sensing system. So this acyl side chain of one of the quorum sensing system, that is a Lassi, is different from the second quorum sensing system, that is RH2. Then you have Agrobacter tumefaciens. Now, Agrobacter tumefaciens, what is it important for? Where have you heard this? You have heard this in the plant transformation, where it forms Agrobacter tumefaciens, as the name suggests. It forms a tumor into the plant cell. Now, this Agrobacter tumefaciens TRA system was used to send the foreign genetic material to make transgenic plant. Much before the discovery of the gene gun, Agrobacter mediated transformation in the plant was very successful. And now also, even now, scientists use the Agrobacter mediated transformation in the plant. Now, this is also a different acyl side chain. Then you have a Vibrio harvey system, where you see that the acyl side chain is very different. But everywhere you have the lactone ring, which is highly conserved. So this is the auto-inducer molecule secreted by various gram-negative bacteria. Now, what is the use? So that's what I was telling you, that how will you appreciate the quorum sensing? You know, that the functionality of the bacteria, how much it is dependent on the quorum. Now, what are the examples of the functionality you can take? For me, it is as simple as the initiating the growth curve, which you can do in the lab by simply using a spectrophotometer. You will realize that if your initial number is not correct, then under any condition, you are not going to initiate that growth curve. Yeah. So even that growth is a function of the quorum. So for example, in case of Vibrio fishery, the end product or the function that it does is a luminescence, that is the production of light. Now, production of the light is something which you can see it, which you can quantify it. That is why it became a very lucrative tool for a lot of scientists to play around with it. You know, and a lot of students, they have developed some beautiful assays to understand how the light production happen and how can you study the quorum in detail. But it does not mean that only light is an indication of the quorum. Aeromonas hydrophila produces proteases. Now, Aeromonas, few decades back, it was simply a pathogen which was hanging around in the environment. But now we know that the Aeromonas hydrophila is it has crossed the borderline in between pathogen and non-pathogen. It is a pathogenic bacteria now. It attacks the lung by producing the proteases, which digest a lot of the ECM matrices which is present in the lung. Agrobacter tumefaciens, conjugation system is successful because of the production of these acylated homoserine lactones. Burkholderia cepacia, a bacteria which requires the production of the siderophores. What are siderophores? These are the small molecules which, when secreted, bring back the iron to the community. That means it binds to the iron and it brings it back to the bacterial community because it is one of the very important metal which is for many of the enzymatic function. Now, Chromombacter violaceum, this produces antibiotic. Irvinia chrysanthemi is a plant pathogen. It is a very strictly a plant pathogen which produces a pectinase. As you know that pectin and the cellulose, these are important component of the plant cell wall, and they are very thick. So if the bacteria needs to enter the plant, they should be robust in producing these enzymes. Otherwise, the entry will be compromised. Or if they are intelligent like Salmonella, where they can take the lateral route entry. Otherwise, like Irvinia, they produces this pectinase. And the production of the pectinase, which dissolves the pectin, allows the Irvinia to get inside the plant. Now, this production of the pectinase is because of the quorum sensing. Pseudomonas aerophaciens, they produce the sphenozymes. Pseudomonas aeruginosa, they produce biofilms. Now, biofilms, again, it has become a talk of every single lab because it is easy to do. The experiments are easy to do. You take the bacteria, you put it in the test tube, just leave it, you know, under certain conditions, you will see a thick coat. Now, this is something which is known as a air-liquid interface pellicle formation or the biofilm formation. And there is a lot of science behind that biofilm. It is just not simply a bacteria which is there, which is standing for long. Now, that biofilm formation is because of the achievement of the quorum sensing, which in turn gives the bacteria a cue that now they should form this biofilm. Now, Rhizobium etli, this is a very friendly bacterium which forms a nodules. You know the nitrogen fixers and the plant benefits from them. So the Rhizobium etli, the number of nodules that it should form is because of the quorum sensing. Yersinia pseudotuberculosis, which is a pathogen, the entire phenomenon of the aggregation of motility is because of the these quorum sensing, acylated homoserine lactone. Now, I had spent a lot of time here just for you to appreciate that every action of the bacteria or every product of the bacteria that you expect can only be successful if the bacteria community is functional. That means it is not a job of a bacterium, but it is a community of bacteria which is coming together because of the quorum sensing and giving you all the products which are very useful for the mankind, and also on the other hand producing a virulence factors which harm us. So both the ways, whatever function it is, you need that quorum. Now, if you look into this gram-negative bacteria, you have all these quorum sensing molecule which belong to the acylated homoserine lactone, but the gram-positive bacteria are very different. Now, the gram-positive bacteria, by the very sheer difference in their bacterial cell wall structure, makes this gram-positive and gram-negative behave very differently. Now, I would just take for granted that all of you here, my colleagues in the audience, know the difference between a gram-positive and a gram-negative. But just in 2 sentences, gram-positive cell wall is extremely, peptidoglycan is very thick. And so they do not have the outer membrane, you know. They only have the inner membrane and a very thick peptidoglycan, whereas a gram-negative, on the other hand, they have a very thin peptidoglycan. So they have an inner membrane, a thin peptidoglycan, and outer membrane. Now, the sheer difference of having a thin peptidoglycan, outer and the inner membrane, makes the entire system of secretion system different from gram-positive and gram-negative. So if you look into these gram-positive bacteria's, you know, the auto-inducer molecule, like Bacillus subtilis or Staphylococcus aureus, you will see that they do not contain this lactone ring like the gram-negative bacteria. They have the small peptides. So here the autoinducers are small peptides, not the lactone ring. So there's a difference between the signaling molecules in the gram-positive and in the gram-negative. What we are going to do is we are going to use a Vibrio fishery luminescence prototype to make you understand the prototype of this quorum sensing system. This system is very simple, and that is why I want to introduce it to you so that you can ask a lot of questions in the end. So I will keep some 10 minutes for the questions so that we can, discuss very well. Now, the Vibrio fishery luminescence system is governed by only two genes. One is LuxI. LuxI makes the molecule. Yeah, it makes this autoinducer molecule. And LuxR is a sensor. That means this autoinducer molecule, it binds to the LuxR. So the moment it binds to the LuxR, this AI molecule and the LuxR complex, it goes to the target genes, binds to the target gene, and then which leads to the production of the enzyme and thereby giving rise to the bioluminescence. You will ask me a question, that is light. What is the importance of the light for the bacteria? In fact, that light is produced as a byproduct in bacteria, but the community and the other small protozoans, they use this light, or the fish, they use the light to hunt for the food. So it is an extremely important for the other, species which is existing with the bacteria. So this is how the gene is arranged. Now, Vibrio fishery bioluminescence, or that blue color, that organ, which is none other than the production of the light. Now, this is the arrangement of the gene. So you see here, there's a transcriptional activator. This is this AI synthase. This is extremely important enzyme, which gives you the oxohexanoyl and homoserine lactone, and the luciferase gene, which leads to the light production. This is the arrangement of the gene. And because of this molecule is produced, and because this molecule is produced, you now have the production of the light. Now, this is a simple system in Vibrio fishery. But when you talk about Vibrio harvae, this is a bit complicated. Now, this is just opposite. Vibrio fishery is always in association with fishes, and they have a symbiotic relationship. But Vibrio harvae, unlike your Vibrio fishery, they do not live in symbiotic association. It is a free living. When I say it is free living, they do not depend. But now the marine microbiologists and a lot of us are trying to understand that it cannot make this 250-kilometer stretch, what I have shown you, that tail of Milky Seas, and never get drifted apart. I mean, how strong the structure is. It cannot even lay down those strong ECM, whatever, not the ECM, but the polymeric substances. There has to be some zooplanktons or some of the structures from the sea, which is making the bed. And they get benefited because the light attracts a lot of food for them. Yeah, but similar to the Vibrio fishery, harvae uses the quorum sensing to control the bioluminescence. And the difference is the fishery is a simple system, but the harvae is extremely complicated. But along with the complication comes the beauty. What is the beauty? It is a golden mine for the researchers or for the students who wants to work on the quorum sensing. And why? I will tell you. First, you can grow them very simply in the lab. You can get it from ATCC or from IMTech Chandigarh, and you can grow them in the test tube. If you have a simple bioluminometer, you can measure the light production. And you know when the light is produced, that means the quorum is achieved. But what is difficult is the genetics of this light production. Now, Vibrio harvae also uses acylated homoserine lactone, but the signal apparatus is consisting of both the gram negative as well as the gram positive. So it can detect both the AI1 molecule and the AI2 molecule. And that is why it is, it is a very important organism for us to proceed in the lab with the quorum sensing system. That means it can take care of the receptor and the signaling by if AI molecule produced by other bacteria, also the AI2 molecule which is produced by the other bacteria. So you can use it as a model organism. Suppose if you are hunting for the quorum sensing molecule, you do not know, you have a bacterium X which you have isolated from the environment. You only want to know that whether this is producing any quorum sensing molecule or not. How will you check it? You may not have any standard. So what you do is you simply take the supernatant and put it in these bacteria and ask a question whether it is producing a light. And if it is responding to any of the molecule, it is going to produce the light. And then you know that, then you go ahead and characterize the quorum sensing molecule which is present in the system. So it produces both AI1 and AI2 molecule, and it can detect both the AI1 and the AI2 molecule. That is why this is a bit complicated when you talk about the genetics of this bacteria, but it is an amazing dream tool for anybody who would love to work in the field of quorum sensing. So now what we will do is we will get into the quorum sensing of Pseudomonas aeruginosa. Now this Pseudomonas aeruginosa quorum sensing, as I mentioned, it is of hierarchical in nature. One is a LAS-I system, one is a RH1 system. Now, why I call it is hierarchical in nature? Because if the LAS-I system does not work, then the RHI system will not work. The LAS-I produces a quorum sensing molecule. This molecule then binds to the RHI system to give another quorum sensing molecule. So if this is not functional, RH will be not functional. Okay, so and how will you understand it? I have made it very simple to understand that how you can study the quorum sensing molecule, because in every lab nowadays I have noticed in the colleges when they come for certain experiments, etc., the biofilm production and its study, it has become very routine these days. It is routine, but the science behind it is extremely complicated. And in fact, we are burdened with all these so many extracts which says that in vitro you can break these, you can kill these, etc., etc. But be very careful when you work with any of these so-called extracts. It can show you some data in your well diffusion assay or in your test tubes. But the moment you try to understand this complicated situation and the community of bacteria, of the bacterial life, everything changes. So it just gets dampened there. So once you understand the community behavior, then you will appreciate much how do you really proceed for the experiment. Now, Pseudomonas aeruginosa, which is also now a very notorious pathogen, it is a pathogen which is found associated with the hospital-acquired infections. It also attacks the lung, it attacks the diabetic foot, and it leads to the chronic non-healing wounds. So it's really very dangerous bacteria now. It was there in the environment for long, but just that it has now turned into a pathogen because of the robust ability to have the two quorum sensing apparatus. So when the bacteria is seated, or it gets settled, it needs a certain number to form the biofilm. So see how it is progressing from the so-called few numbers to the biofilm stage. Now the biofilm is nothing but a community of bacteria which lays down certain molecules. You call it as the extra polymeric substances, or the EPS. And the biofilm is laid during many stages of the life of bacteria. So suppose if it want to attach and invade a host, or it would like to stay together near the root, they will form a microcolony, and then they will lead to the biofilm. Now, bacteria should never be always be in the biofilm. So there is a making and breaking of biofilm. The community or the crosstalk between the bacteria is extremely important to make them form a biofilm. And similarly, when they want a cue that now they have to break the biofilm and get into the planktonic phase, they have to break it. But what you understand from here is that this system uses the LAS-R system first to make a certain AI molecule, which leads to the production of the toxin like pyoverdine and then elastase and other toxin A. This mounts the host attack. That means the cells are actually undergoing a lot of, the mammalian cells are undergoing a lot of stress and they are almost dying. But then the same AI molecule now kickstarts the second quorum sensing apparatus that leads to the RHIR, and this second quorum sensing apparatus leads to the production of many virulence factors, including pyocyanin and biofilm production. Now, pyocyanin is a toxin, but the beauty of the pyocyanin, it is blue in color. So when you grow Pseudomonas aeruginosa, and if both your quorum sensing apparatus is very intact, then you will see that your solution by eyes, they are turning blue. So instead of using a bioluminometer, which is expensive for Vibrio harvey, you can use Pseudomonas aeruginosa, because you can use a simple spectrophotometer in the visible range and calculate the amount of toxin produced, because it turns blue. And if it is not turning blue, that means your quorum is not achieved. Yeah. And then it mounts the host attack. So both of these, LASI system and the RH system, both of them, they together work to mount the host attack. And that is how the host then paralyzes. Now, here is a very important point which I would like to point out before I open up for questions. The biofilms are extremely resistant to the antibiotics. What is the reason? Because all these thick extrapolymeric substance that it produces does not make the antibiotic to permeate inside. And that is why you can dump the system with micrograms, milligrams of antibiotic. The bacteria, it will not happen anything to the bacteria. And at the same time, the bacteria gains the so-called antibiotic resistance. Now, for decades, we did not even understand that this communication is so important. And we went on treating with higher and higher and higher doses with new generation of antibiotic. Only now we come to know that now we are in a big soup, because now it is all the XDRs and the MDRs and the so-called superbug. Now that what we give the threat to the bacteria, they are going to come up with a much bigger threat. And if we do not understand the language of communication, or if we do not understand that the bacteria works as a community and not as a single bacterium, we will in future also end up in having a very terrible time where the infectious disease cannot be wiped out. Now also it cannot be wiped out because bacteria life will never end. Yeah. So having said that, I end my talk here, and it is now 11:56, and I will now open it up for questions. Colleagues, you kindly ask all the questions that comes to your mind. It can be, anything, right from as simple as what is a bacterial cell wall to what you meant by communication. Don't have a fear. You can, no questions can be stupid. So please make sure that you just discuss and let us communicate now.
Ma'am, one question from chat box, ma'am.
Yeah, I will read the questions. 1 minute, and I hope that there are a lot of questions in the chat box. Okay, and if you are shy, kindly write it in the chat box, but don't be shy. It is good to have. Okay, now let me read the questions from the chat box. Okay. Okay, so where is the?
Shall I read, ma'am? Shall I read the question?
1 minute. I'm somehow not able to see because, okay, what type of quorum sensing signals are used by gram-positive bacteria, right? So, just 1 minute. Hello? Yeah? Yes. Yeah? Line is working. Thank you. Yeah, thank you. Okay, sorry. So, yes, who asked this question? And thank you for whoever has asked. So I'll answer that. Now, as I mentioned to you that the quorum sensing molecule is very different in gram-positive and gram-negative. In case of gram-negatives, it is acylated homoserine lactone. That is an auto-inducer one, and its variation. In gram-positive, it is a short peptide. So these peptides, they are not named like homoserine lactone. These are very short stretch of peptides. So now what is very interesting with the gram-positives is a lot of these molecules are secreted, and it can be one of the simplest example I will give you is a D amino acid. Now there was a beautiful paper in Science which came up showing that how D-serine, that is a D amino acid, D-serine, in fact leads to the disruption of the biofilm when it is present in the environment, in the bacterial community. So as simple as a D amino acid, and I think colleagues, that how D amino acids are very, special to bacteria. We don't make D amino acids, right? We all have L form. How bacteria make D amino acids? Because they have the enzyme known as a racemases or racemases. So they convert the L form to the D form. So here it is a short, small peptides and some of these D amino acids. And now the quorum sensing hunters are hunting for many of them. Anything which is dependent on the community or the density dependent molecule, you term it as a quorum sensing molecule. Now it is up to you how you go about purifying them. HPLC purification, then identifying it by mass spec, many steps involved. But if you are lucky, you hit upon that molecule, and then that becomes a very important quorum sensing molecule. Yeah. So next question is this quorum sensing same as that in the honeybee? Okay, so now quorum is a ubiquitous phenomena which is there in every single living organism. Be it in honeybee or be it in human or be it in animal herd or anything, be it in birds. The question is about the signaling. Now in honeybees, yes, you are right. Who asked this? Shruti. So Shruti, yes, it is the honeybees uses a very different kind of the quorum sensing molecule, some different volatiles, which it senses to have its surrounding, whereas the bacteria use acylated homoserine lactones, many of the shorter peptides, and many other molecules to cross-talk between each other. The language will differ, but the phenomenon of community behavior remains the same. So yes, it is very much true. Kanaga, Durga, good morning. This is Kanaga. Thank you, Kanaga. You are a student of biochemistry. There is no evidence that Vibrio fishery cause disease in human or other organism, but it does contain a homologue of the toxin make a disease in human. The point here, cholera is found in association with the fish. Now you can ask, what if you eat the squids and the deep sea fish? You know how the Vibrio cholera acts? Now Vibrio cholera is a very humble pathogen, I would say, because Vibrio cholera causes the disease cholera or the choleric dysentery because it harbors a bacteriophage. You know, if you get rid of that bacteriophage from the Vibrio cholera, the disease will not be caused because the toxin, the cholera toxin, is encoded in that phage. Now, the moment the Vibrio cholera attaches to the epithelial cells, it secretes the toxin into the epithelial cells. Now, Vibrio fishery, even if it is eaten up by the fishes, is going to get into the lumen and get chewed up and get digested. So there is absolutely no evidence, and also there are no reason that why this Vibrio fishery will cause the disease. And Vibrio the cholera, by the way, it secretes this toxin, which is called as a cholera toxin, with the help of a secretion system, which belongs to the type 2 secretion system. It's a very interesting. Sometime if you get a chance to come to your college, I will explain you about the secretion system, because it is a secretion in bacteria. It is a sophisticated system which takes the protein which is made in the cytosol all the way to the destination, you know, be it the host cell or be it in the environment. And it is not a very simple process of simply diffusing out. Always remember, colleagues, bacteria is a very sophisticated machinery it possesses. Nothing is simple or porous. So everything is very well controlled. So yes, it does not cause any disease, but you asked about the possibility. There is no, I would never say not possible, because in science anything is possible. Nobody has done a study to isolate that bacteria and then put it in the host to ask a question whether it can lead to a disease in human, in a host too. Human, you cannot do it. You need to rely on certain outbreaks, but till so far there is no outbreak by fishery. And Dr. Abhilash, I am from humanities background, curious to know about any stream of science. That's very nice, because I encourage that. My question is, can science and humanities converge to find ethical universal application of such advanced science to the benefit of the poor in transforming their access to the better treatment that is cost effective? Yes. First of all, Dr. Abhilash, the most important point here is that how have we gone overboard? It is not a fault of anybody, neither a clinician nor a researcher like us who are always in a warfare to curtail. Now you are a professor of humanities, and you know very well that if you understand if you honor the origin of life, the life of an ant or a honeybee or a bug or a plant is no different from us, right? How does it matter to the universe that whether it is an ant or a mosquito or a bacteria or a human? For that, everybody is equal. Now, if the equality comes in, that the right to live comes with everybody, it is because now the mankind is suffering from a disease, bacterial disease or viral disease, we are cursing them. And the reason why this happened, and it is going to happen more, is we have entered into a warfare. What is that warfare? That warfare is creating a weapon. In this case, the weapon is antivirals, antibacterials, thinking that we are saving human life. Maybe for a short term, it looks good because the lives are saved. But is that life safe forever? That person who feels okay for some time actually suffering from a lot of extra complications and making the life much bitter in the end stage. Similarly, to counter the weapon that we are creating in the name of antivirals and antibacterials, the bugs are becoming superbugs, and because it is their right to live. Now, how do you stop the warfare? We have to come to a halt now in understanding the community. Their community is extremely strong. The simple, whatever new generation antibiotics or antiviral is not going to stop them. How do you stop them? Now we have to come to a harmony. And that harmony is precautions. That means you do something that you don't allow the bug to get inside. But when you allow and the bug is inside, do not treat it with such a harsh antibiotic, then you are making it worse by creating antimicrobial resistant and becoming superbug. So that harmony is a branch which should now be inculcated. Humanities can do that because you have that strength to make people understand that how important is the harmony. Nothing can be fought with war. We cannot fight the war of bacterial disease or viral disease with antibiotic and antivirals. We have to simply first understand and then come to this harmony to make sure that none of us are hampered. Yeah, so cost effectiveness, medical science is becoming very vicious. Though I would say that the motive is to save the life, but I am not very sure that how this is being implemented and how much we ourselves are becoming really hyperactivated when it comes to treatment. I mean, are we doing right in terms of really going to that kind, even if it is a small thing? So we need to be intelligent, I think, to understand what is going on and then take our action accordingly. Cost effective, yes, it should be made because that is our, again, a right to health. And under that right to health and right to live a good life, it is not with a high morbidity and high, a life which you cannot even enjoy. So that kind of a good life is something very different, which now then we need to go back to our villages and ask a question that why everybody is so happy there. Anyway. Okay, now the next question is from Dr. Jayaprakash. Thank you, Dr. Jayaprakash. How can quorum sensing solve antibiotic resistance? Very important question. We ourselves, we are working on it, just that we have done so much harm to them by creating so much of antibiotics, new generation, and their community have become stronger. Now we have to understand that how their quorum sensing molecules are produced when the antibiotic treatment is given. Why they are generating these XDRs and MDRs? Because the community is so close. What is that molecule? Now, if you identify that molecule and then you try to deplete it from the system, which is known as the quorum quenchers. So many of the groups, they are also actively hunting for quorum quenchers so that you do not allow the bacteria to cross-talk, and then maybe the antibiotic can act against them. Now, how quorum can solve the antibiotic resistance? I would simply say that first, stop using the antibiotics once for all. Only in the case of very severe infections, one has to use it. and that too not for very small infections, because the moment it is done, even quorum sensing inhibition will not work, because even the bacteria, the planktonic, form, after some time will understand that they are getting a big threat, and then they are going to develop that resistance. But the first thing is not to use the antibiotic at all. And even if you're using it at some of the really later stage, you make sure that you use in a combination with a quorum sensing quencher, so that you quench the quorum sensing, the bacteria do not sense, they cannot make the biofilms, and then the antibiotic will act on them. Yeah. Okay. Challa is saying, send me feedback link. Okay, Chala, you will be getting the feedback link from the organizers. Yeah, so organizers would love to have the feedback, so kindly give them. As Sacred Heart College has taken so much of initiative to organize this, and so kindly give the feedback. Okay, Dr. P. Srinivasan, thank you for asking question. Are there any mimicking molecules similar to QS molecule? If there's, then what are their applications? Yes, Dr. Srinivas, there are molecules which are, similar to QS molecule means that if you have the natural QS molecules in the system, if you are lucky to identify, like Eberhardt or Engelbrecht, they identified the acylated homoserine lactone, or many other groups are isolating a small peptides from the gram-positive bacteria, or the D-amino acid, as simple as D-amino acids from the gram-positive bacteria as a QS molecule. The mimicking molecule means nothing but you can synthesize them. The sigma gives all of these molecules in a pure chemical form. If you buy those chemicals and you put it in the bacterial community, you will see that they kickstart their process. So yes, there are similar molecules. See, the question become very relevant. Maybe in host, mammalian system, whether they make any molecule which somehow mimic that of the bacterial QS molecule, and within the host, if the bacteria somehow shut off to make their own molecule, whether the host molecule now can direct the bacterial community. It's a very good question, but at the moment I have no answer to this, because I am not aware of any of the host molecule which can trigger the quorum sensing communication in bacteria. But I have no doubt that there will be some, because the bacteria in the host is very different from when the bacteria is in the environment. So I am sure. Mary, thank you for asking. Quorum sensing is intraspecific communication. Is there any type of interspecific? Oh, quorum sensing is both intraspecific as well as interspecific. Both of them act, because you know what happens in the gut. In the gut, you have 230,000 different species, and they communicate with each other. So this, say E. coli communicating to E. coli is simply intraspecific. But if E. coli communicates, say to Bacillus subtilis, it is interspecific. Or say Vibrio cholerae, which is a pathogen, has to communicate with the gut flora and fool them, because otherwise it cannot reach the intestinal epithelial cells. So there are both interspecific as well as intraspecific communication, both these AI1 and the AI2. Actually, AI2 is the interspecific communicator molecule. Yeah, AI1 is mostly for this intraspecific. So yes, there is both interspecific as well as intraspecific. Pooja, thanks Pooja for asking. Pooja Chaudhary is asking that as you say that they work in community to cause disease, similarly, is it possible that they cured the same disease with the help of the community once the antidote for the disease is designed? Okay, I'll give you this example, answer this question in a different way. They definitely work in a community to cause the disease. Similarly, your gut flora works in a community to stop any of the gastrointestinal pathogen to actually get inside, Now, it is really a community behavior to do whatever they design. But suppose if you're asking the question that the same pathogen that is causing a disease in a community, whether they can cure the disease with the help of the community once the antidote for the disease is designed. Again, suppose if you have a Salmonella, say Salmonella typhimurium or Salmonella typhi, which is a human pathogen, or say Vibrio cholerae or Mycobacterium, which is a pathogen. Now you have an antidote for another group of bacteria, which can outcompete this? It's a very good question though. In vitro, there are a couple of data which suggested that if you pre-treat your cells, say by lactobacillus, rhamnosus, or Saccharomyces boulardii, or by bifidobacterium, you do not allow the pathogenic bacteria to attach to the cell. That means they are outcompeting. That means the antidote actually works. But it is also, there are a lot of trials that had happened. It's a double-blind placebo-controlled trial where they, to stop the infantile diarrhea by rotavirus, they fed the infants with Saccharomyces boulardii or Lactobacillus rhamnosus, which stopped the bacteria, the infantile diarrhea. That means, yes, there is an antidote which is available of a bacterial species or yeast species, which can outcompete your disease-causing pathogen. This is possible. It's just up to us now to, figure out how do you identify each antidote and then try to develop that model. Okay, Survachala Rajalakshmi, thanks for asking. And the question is quorum sensing limited to bacteria only? Yeah, it's a good question. Wherever I go for the talk, the students ask the same. It's a very important question. No, it is not. Now, one very bright young student asked me that, does virus not have quorum? See, virus is in the borderline, we say, it's in a layman's language, in the borderline of living and non-living. Outside, they just cannot grow, but they need a host. Within the host, how do they, decide that should be the viral number before they burst out? If it is a lytic virus, they come out or they bud out. Yeah. There are beautiful papers which are coming up that actually there is some sort of quorum which also operates in the virus. So similarly, no, it is not related at all to, it is not limited to bacteria. It is there in every life form, in every single life form. It is only up to us now to understand how do you, in protozoa, say you're studying Leishmania infection. It is just not that one Leishmania which is causing, right? How is the crosstalk between that Leishmania? They are protozoa, they are bigger than the bacteria or the viruses, and how do they? So there is a communicating signal. It is not limited. Thank you, Dr. Prakash. So you got the answers. Thanks. And Dr. Abhilash, explanation was thought catalyzing. Thank you. Yes, Dr. Abhilash, you play a major role because you're from humanities. And remember, now, even in medical schools, they are trying to introduce the subject. In our UG course, we have a humanity as one of the mandatory courses. It is just for us to understand that how each life form is equal, and that is absolutely just not because that we are so-called talking and we are little bigger means nothing to the cosmos or to the universe. It means nothing. We are as equivalent to ant or mosquitoes or bacteria or viruses. So humanity can only teach that. And if we are taught, we will be grounded. All our egos will disappear and we will be grounded. And we will know that we have our boundaries. Let us not act big. So kindly play that role in, in making everybody grounded. Humanities can only do that. Kasturi Harrison, which one are QS autoinducers and QS inhibitors? In this particular class, because it's very limited time, what I told you, they are all QS autoinducers. I have not talked about the QS quenchers or the inhibitors. There are many molecules, like I was talking about D-serine. Now, D-serine somehow act as a QS quencher because the D-serine breaks the biofilm. Not only D-serine, there are a couple of other amino acids which breaks the biofilm at certain concentration. So how does it do or break the quorum? There are a lot of mechanistic explanations available, but not of this particular only D amino acids. How do they do that? I mean, you have a preformed biofilm, you add something, it just breaks. Similarly, lot of these, I am seeing a lot of proposals with this herbal extract, which say that when they put it, it is breaking. These are, you put certain, say, some extract, it breaks the biofilm, that they comes to the planktonic. These are all explanations to go ahead and fish out that molecule, which will break the community behavior. So in this class, it are Kasturi, it is all about quorum autoinducers. I have not talked about the constant, but the acyl change. So yes, different conditions will differ. Yes. How will they communicate with each other? Good question. Now, receptor or so both molecule.
Ma'am, sorry for interrupting. There
Interspecific.
is a voice breakage from your side, ma'am.
They
Voice is not clear.
receptor. Yeah. Receptor. This is the receptor. And then it gets into the downstream. Extra therapies, very good. They were very different because their cognitive behavior is very different.
Pungode, ma'am.
Yes, sir. Here only, sir. Ma'am will left, sir.
Ma'am was done. Okay, so maybe some network
Ma'am
issues.
rejoined now.
Rejoined, okay. Okay. Yeah, sir, I think she's having some issues with the voice.
Sorry, colleagues, I got logged out. Just 1 minute. Oh, why the chat? Kasturi, could you hear my answer?
Yeah, ma'am. Yes, ma'am. Yes, ma'am. Thank you so much, ma'am. The voice is breaking because of network problem. It might be.
Can you hear me?
Yeah. Now
Yes,
I can
ma'am.
hear you, ma'am. Now I
Hello?
can hear you. Now I can hear you, ma'am.
You can hear. Okay,
Yes,
but
ma'am.
could you hear the answer? Okay,
Yeah, you are saying
okay.
about the auto
So I think I was on Dr.
inducers. Yeah, yes,
Autoinducer.
ma'am. Ma'am, as I'm dealing with the healing process of a wound which is related to all these microbes. So that's
Okay,
why I
okay.
wanted to know about the inhibitors also, ma'am. It
Yeah,
would be
yeah,
fine
that's
if you
what.
have
So the
another. Yeah.
yeah, so in this platform, it is not possible, Kasturi.
Okay. Okay,
Maybe
ma'am. If you,
you write to me at deepa@isc.ac.in. I'll send you some papers on the quorum quenchers. Yeah.
okay, ma'am. Okay, ma'am.
You can
Okay,
get a very good idea about how do you first break it, and then you can treat it with a low dose of antibiotic. Yeah.
okay. Okay. Thank you, ma'am.
Okay. So
Please provide the email address there in the chat box, ma'am. I could.
I will type it. Just 1 minute. Let me type it.
Thank you so much, ma'am. Thank you.
deepa@iisc.ac.in. Okay, here you go. So all of you kindly feel free to write to me if you need any papers or if you want to ask more questions. So Bhuvanesh, your question is answered. Bhuvanesh?
No, ma'am. No, ma'am. No, ma'am. That is did not answer to you.
You did not get my answer. You did not hear it, huh? Okay,
Yeah,
I
ma'am.
will answer to you. Yes, what you said is correct. There are different group of bacteria, and what is very important that how do they signal each other is through the receptor. So the receptor will recognize that signal And you know, the AI2 is interspecific communicator, no? Because it can talk to many bacteria. How do they receive the signal? That is with the help of the receptor, which is present. Now, when you talk about thermophilic bacteria, these are very different range of choromolecule, as you know, because of the sheer temperature. So, you know, 100 degrees, 150 degrees, extremophiles, etc. And they have a very interesting set of the molecules which are used as a quorum communicators. Yes, so because of the receptor, that diverse receptors, they can sense it. Is it clear now?
Yes, ma'am.
Okay.
Thank you.
Okay. There are no more questions, but it was very nice. All of you could write all the questions in the chat box, and all the questions were very good. All the questions were very valid, and I had a very wonderful time discussing with you. So thank you very much for everything. If you have any burning question, please feel free to talk if we still have time. Dr. Niyaz Ahmad and Dr. Gomati, is there time or there is a second speaker already ready?
Actually, the second session is also given by you only, ma'am.
Is mine? The second session is
Yes,
mine?
ma'am. Yes, ma'am.
Okay, and that session goes. So do you all want to take a break or shall I continue?
What is your opinion, ma'am?
No, how about, let me see what our participants feel. Do you want to take a small break or shall I continue? Just write in the chat box. What do you feel? You want to take a 5 minutes break?
But that is not possible for the audience. They will not
possible,
immediately
then
enter.
Let us start.
Yes.
I hope that all of you
are you ready,
still,
ma'am? Do you need any more refreshment, ma'am?
I don't need, just one call I will attend.
Okay.
Hello. Working. It's working. Thank you. Yeah. No, I don't need anything. So if the colleagues are ready, I am happy. I'm very happy to talk with them. So then let me continue, right? I will continue because now what I'm going to tell you is really very interesting. I'm going to introduce you. So Dr. Gomati, now I continue till 12:30, right? Hello?
Yes, ma'am. Yes, ma'am.
Till 12:30. Okay, great,
Yes,
great. So colleagues, now what I'm going to talk to you, I'm not very sure whether you have heard about it. I'm going to discuss with you a concept of a programmed cell death. I do not know, and also just keep, okay, please continue, ma'am. Okay, great. You all are happy to listen to me and I'm happy to talk to you. That's great. So the concept of the programmed cell death, which I'm going to talk to you now, is something in bacteria that we cannot digest. We know about programmed cell death in mammalian system, and you call it as apoptosis, right? But what is this programmed cell death in bacteria and how it is related to these community behavior or quorum? So let me again share the slide and continue from here. You can all see my slides, right?
Yes,
you can all
ma'am.
see my slides?
Yes, ma'am. Yes, ma'am.
Okay. Okay, now I will also still not put it in the full screen mode because there is some problem with the bandwidth. So the full screen mode is not visible as long as good enough. Okay, colleagues. So just refresh your mind in a way that you now digest the fact and appreciate the fact that what is the reason that we cannot get rid of the bacterial community. This will be a mind-boggling change in your mind. First, to understand two concepts. Is it really true that what we have learned about the action of the antibiotic is the only way by which bacteria are killed, or there is something else to that? Okay, so just listen to the story and at the same time start typing your questions in the chat box as and when it comes to your mind, so that at the end of this, we are going to have a wonderful discussion like we had in the first session. What do you understand by the programmed cell death, first of all? Now, the PCD or the programmed cell death is nothing but a death process in a community. Now in mammalian system, you call it as apoptosis. The very fact that our fingers are separated is because during development, these cells are apoptosized so that we can have free digits. You might have seen people where in the leg, these two are kind of joined. That means the developmental defect. There is no apoptosis that has happened. So the process of apoptosis is a programmed cell death in the mammalian system. Now, what is it about that in the bacteria? I mean, how bacteria can sense programmed cell death? To answer to this, and now we know that this is the only reason, and it is an eye-opener for us to understand that how we should not be overdoing with anything to harm any community, be it a pathogenic bacteria. Now you see what is going to happen. Now, the good example that I can take you here to teach you or to tell you about the programmed cell death is a toxin antitoxin module system. Okay. Now, this toxin antitoxin module system is a MAS-EF system, and that is there in E. coli. So, E. coli is a very simple model, and the E. coli bacteria it's like elephant, of the lab, in the sense that what can happen is true in elephant is true in E. coli. That beautiful model, the E. coli is, in which you can do many things. You want to study aging in bacteria, you use E. coli. You want to study the death, programmed cell death, you use E. coli. So here we are going to discuss using the toxin antitoxin module. Now, what do you understand by the term toxin antitoxin? That means there is a toxin which is there in the system, and there is an antitoxin in the system. Now, this toxin is not for killing mammalian cell or other. Okay, this toxin is a self-harming toxin. You will also ask a question: why a bacteria should have a self-harming toxin? It's like we said, no? Why? So anyway, we will discuss that why this is important and how it protects the bacterial community. Now, the toxin and the antitoxin, it is always in a stoichiometric ratio. That means the toxin is bound to the antitoxin and is never free. Yeah. And this toxin antitoxin module is encoded by the gene MAS-EF. Okay. Any thing which you cannot understand or any problem, just keep typing in the chat box and I will come to all of them. So this MAS-EF system is an example of toxin antitoxin. It's a module. These are the two genes. One makes a toxin, one makes an antitoxin. And when they are made, they're always in a stoichiometry. So the bacteria is happy. It is not self-harming when they are in combination. What is this antitoxin here? Now the antitoxin is encoded by MAS-F. Okay. And the toxin is encoded by MAS-F, and the antitoxin is encoded by MAS-E. Now, this toxin, which is MAS-F, suppose if it is set free, suppose due to some reason, and the reason we will come later, it is set free, it will go and cleave the mRNA. You know the mRNA, which is very important now to make your proteins. It will cleave or chop off the mRNA at the ACA sequences. Now, ACA sequences can be there so many places in your mRNA. What is the specificity? So there is a certain hairpin loop. When you have the ACA sequences in front of that, it will go and chop it off. So what happens when this toxin chops off this mRNA? And that is why it is an endoribonuclease, because it chops off the mRNA. When the mRNA is chopped off, what will happen? Protein will not be formed. If a protein will not be formed, what will happen? Actually, the bacteria will undergo death, because these proteins are essential for the life of the bacteria. Now you have the two situation. You have a toxin antitoxin, which is always in combination in a module, and the toxin is not set free. That is why the bacteria is happy, it is growing. But the moment your toxin is free, it will go and chop off the mRNA. And this is coded by MAS-F. Antitoxin is coded by MAS-E. Okay. Now, MAS-E encodes a labile antitoxin. What do you mean by a word labile? Labile means sensitive. What it is sensitive to? Now, this antitoxin is also a protein. Okay. Toxin is also a protein. Now, this antitoxin is very sensitive to the clip protease, which is produced by the bacteria. So under certain conditions, which we will come across very soon now, whenever there is a production of these antitoxin, along with that, there is a high production of the clip protease, which will cleave this antitoxin. But the toxin is resistant to the action of the protease. Then what will happen? Your toxin is set free. The moment the toxin is set free, it creates a havoc in the cell. Imagine if this particular process of toxin antitoxin should have been active, bacteria would have become extinct by now. It cannot become. So this toxin antitoxin module is a stress induced phenomena, and it is a stress induced phenomena which is dependent on quorum sensing. How it is dependent on quorum sensing, this we are going to understand very soon, and how it is a stress induced phenomena. Okay. So the stressful conditions in a cell, in a bacterial community. Now, when I tell you a stressful condition, I am telling you a stress which is given by antibiotics. As you know that antibiotic or the AMR or the antimicrobial resistance has become a talk of the world. You take WHO, you take CDC, you take any journal, any paper, newspapers, they only talk about the superbugs, the generation of MDRs and the XDRs. Who has led them to this condition? It is we ourselves, because we have not understood enough. Anyway, so in this particular understanding of this topic, I'm going to take the example stressful condition of only antibiotics. Okay. So any antibiotic which you give to the bacteria, be it ampicillin to rifampicin to trimethoprim to nalidixic acid, irrespective of their action, be it binding to the gyrase and doing a certain stopping in the DNA transactions, or be it protein synthesis inhibitor, or be it a peptidoglycan cell wall cross-linker inhibitor, irrespective of its action, colleagues. So remove from your mind the classical action of antibiotics. Yeah. It can be any antibiotics. It's irrespective of their antibiotic action. Any stressful condition, when it is given to the bacteria, there is an expression of this massive system. That means the toxin and the antitoxin system expression goes high. Now, along with the toxin antitoxin expression system, the clip protease, that expression also goes high, which will then cleave the antitoxin. That is a mass E, which codes for the antitoxin that is cleaved. Okay. Then what happens? The toxin is free. This toxin, which is set free, acts on the mRNA with that particular ACS sequences. And this leads to the death in a population, bacterial population. Now, this death is mediated by a quorum sensing molecule, and this quorum sensing molecule is known as a EDF, that is extracellular death factor. Okay, now the mass EF mediated cell death is a population phenomena. Now, many of you who would like to understand more on quorum sensing and the quorum sensing processes, you can call anything as a quorum dependent if that activity is dependent on the population or on the density. That means that when you perturb the density or when you change the number, that particular process is perturbed. Then you know that this is dependent on the population. When it is dependent on population, that means it is dependent on quorum, right? So it's an easy way to explain. So the death in the massive system is a population dependent phenomena, and this entire death process is because of production of a molecule which is dependent on the density, which is known as EDF, or extracellular death factor. But for you to understand or comprehend everything in the mind, let us do, let me explain you a very simple, elegant experiment that was published in Science, which makes these experiments, to make us also believe that it is always the simplicity and the novel ideas which leads to the bigger discoveries. It is not you begin with next generation sequencing and then only science can move, or you need to have a knockout mice, then only science can move. No, you begin with that very novel question. The science starts with the novelty and the naiveness of the area. what you ask, then you develop it into a science. And this talk will tell you, or this experiment that I'm explaining will tell you that this is a science paper which they did with a couple of test tube and agar plates. But what was very interesting was the idea. Okay, so in a nutshell, what happens? Whenever there is a stress to the bacterial community, in this case, the stress can be by any antibiotic, there is an induction of the MAS-F. Because the MAS-E is degraded, the MAS-F is free to do the problems in the cell. Now, whenever there is an induction of this toxin, the toxin will cleave the mRNA at the ACA sequences, which will lead to the global inhibition of the protein synthesis. But this is not the end of the story here. In a very small population of the bacteria, what happens that there is a differential induction of the set of gene, which will make the bacteria survive, or it will lead to the death. Now, this death and this death is same. It's just that this particular death is very fast. It is very rapid. So bulk of the bacterial population is dying, and this death is a bit slower. Here, the catch is, and now you know that why we cannot get rid of the bacteria, is this surviving population. You can dump, one can dump oneself with antibiotic, but you cannot get rid of this survival population. Why you cannot get rid of this survival population, we will come very fast. So now this MAS-E, MAS-F mediated death is a population dependent death, and that is why it is a quorum dependent programmed cell death. It is not a random death that you give the antibiotic, they are dying. And that is the only reason now, if you look back, many a times your antibiotic resistance, the sensitive assay never works, because you're thinking, oh, all my bacteria should be killed. Why am I getting colonies still? Has it become resistance? No, it is a property of a bacteria to survive, because it is programmed in such a way that a small population will survive and the majority of the population will die. Now, let us understand this entire pathway with the help of a very simple experiment. And that simple experiment goes like that. You can do it in your lab too if you have a very simple tool. Now, long back, when you want to study that whether my gene is active or not, or whether it is expressed or not, you resort to making a genetically engineering construct, right? And you use the lac operon, because it's a very well-defined operon. You put your gene of interest in the lac operon and use that lac operator promoter system to induce your gene of interest. In this particular system, look at this, just figure A. This is an assay to tell you that the newly synthesized protein in the cell is happening or not happening. And how do you understand that? What happens when the proteins are getting synthesized in the cell? One of the soluble tracer that you can add to mark the synthesis of the new protein is the amino acid methionine. So you add a radioactive tracer, S35. S35 is a beta emitter. So the methionine is labeled with S35 and you put it into the medium. And when the S35 is incorporated into the newly synthesized protein, you can measure the incorporation of the S35 methionine by using a radioactive counter. So you take the total cells, you precipitate, and then you go to the counter, radioactive counter, GM counter, and then it will give you counts per minute. So what your y-axis is showing is the count per minute of the incorporated radioactive methionine into newly synthesized protein. And your x-axis here is showing addition of IPTG. Now, IPTG is nothing but isopropyl thiogalactoside, which is an analog of lactose to induce the lac operon. Now, in this particular E. coli system, they have genetically engineered the lac operon, expressed the toxin, that is a MAS-F, and used IPTG as an inducer. Yeah, I hope you got the system right. Now, the moment you induce the system with addition of the IPTG, this is known as the inducible system. Okay. Until and unless you add IPTG, the toxin will not be formed. So you see that when there is no IPTG, there is no toxin formed. You have a beautiful incorporation of That means all the proteins are nicely getting made. The moment you add IPTG, that means the toxin is made. And what happens when the toxin is made? The toxin is the endoribonucleic. Go and chop off your RNA and your protein synthesis will come down. That means your S35 methionine incorporation will become low. And that is what exactly you see here. The moment you add IPTG, your S35 methionine incorporation is low, and otherwise this is very high. Now, suppose if the authors were very boring and they would have told, oh, it is nice, I am at least getting low S35 methionine incorporation. That means toxin is acting and I stop here. They would not have made this as a science paper. They saw and they very carefully observed that the S35 methionine is lower, no doubt, but it is not zero. What does it mean? It means that there is some amount of S35 methionine incorporation going on. That means some subset of the proteins are being made. Now, what is this subset of protein? And why is it being made when there is a stress, when there is antibiotic stress? That led them to take this lysate and run a gel. Now, all the proteins which have incorporated S35 methionine, you can do a process which is known as autoradiography. That means you run an acrylamide gel and you expose your X-ray sheet. You put it, you nicely put it in a cassette and keep it in the cold room. And after some time, you develop that X-ray sheet. All the radioactive labeled proteins will be showing you like this kind of bands. Okay. Now see, when you do not have IPTG induction, so many proteins, newly synthesized proteins, because the toxin is not there. And the moment you induce the toxin with the addition of IPTG, almost it became white. The background is white, but it is not zero. There are proteins, very distinct proteins, which are being formed. You know, what are these distinct proteins? These are little low molecular weight proteins which are being formed. Now the question is, in spite of having the toxin, why is this protein being formed? What is the rationale of making this toxin? So this is known as reverse genetic. When you want to know that why is this protein being formed, what you will do is, and who is making this protein? You know the protein, but you want to know which gene is making the protein, right? You are going to excise this band. You will know the amino acid sequence. From the amino acid sequence, you will know the RNA sequence and then the DNA sequence. Okay, so this is known as reverse genetics. And you exactly know. So that is what they did. It's a huge amount of work though, to figure out that all these bands are corresponding to what kind of protein. They fished out the gene and then they knew that which gene in E. coli is making this protein, and then they made a knockout of the gene. Okay, now colleagues, don't get worried about so many nomenclatures here. I will make it very simple for you. This test is in E. coli again. This is known as a viability test. What does it mean? That means that you give E. coli any of the antibiotic, be it spectinomycin, be it nalidixic acid, and you give it. So concentrate on this. This is the wild type with spectinomycin with nalidixic acid. And what you see on the right hand side are the survival genes, named as the survival genes. And this is named as a death gene. And how did they score for this? It is very simple. It's a plating assay. You expose them to the antibiotic and you plate them. And then you calculate the percentage viability by counting the colonies. It's as simple as that. Now understand what is happening. The moment you are making a knockout in these genes, MASI-MASF, this is the clip protease, which I told that gets enhanced when you give them antibiotic stress, and this clip protease will degrade your antitoxin so that the toxin can act. Okay. Now, all of these genes, when you make a mutation in E. coli, it's a very simple way to delete the gene out. You see that these ones do not die in the presence of antibiotic. They do not die. MASI-MASF is the control, because this MASI-MASF is the toxin antitoxin module. Okay, you concentrate on MASI-MASF only. I don't think that you should concentrate on here, because that becomes too much to knowledge to comprehend in the mind. Just think of massive system. Now, the moment you delete this toxin antitoxin system, which behaves in a population dependent manner, you see that they become unresponsive to the antibiotics, and their viability is almost more than 80%, 90% in spite of the antibiotic. Then you will ask a question, what happened to the traditional role of antibiotic here? That means, yeah, whatever we have studied in the textbook, the conventional action of antibiotic, there is something much prior signaling happening, and that conventional action may not be even true when it comes to the action of antibiotic to bacteria, with bacteria, and no doubt that the bacteria are simply not getting extinct because it is a quorum dependent phenomena. Now, if you look at the survival genes, now all these genes which belong to the survival gene category means if you delete these genes, the bacteria cannot survive in the presence of the antibiotic. So these are termed as survival genes because they cannot survive. These are death genes. That means that if you delete these genes, the death will not happen. So these genes are responsible for the death, and these genes are responsible for survival. I hope that these are clear to you. Only concentrate now on MASF, because that is that module that I started with to make you understand about the programmed cell death in bacteria. Okay. So now this is with spectinomycin phenylidic acid. The same thing can happen with trimethoprim or with any other rifampicin, any antibiotic that you may take the example of. So the thing is that how the bacterial community is understanding now. It is a community of millions, billions. How is it understanding this density dependent phenomena? So just concentrate on this for your own understanding. I made a schematic which will be very beautifully, you will be able to understand it. Now, you will trigger this MASF system by a stressful condition. And what can be the stressful condition? The stressful condition is addition of antibiotics, okay, in this case. Now, the moment the bacterial community faces that antibiotic, there is a upregulation of this MASF toxin antitoxin module, and also the upregulation of the protease. Now, the protease will cleave the antitoxin, which will release the MASF, which is a toxin, okay, from the inhibitory effect of antitoxin. MASF, being a toxin, is going to act like because it has an endoribonucleolytic activity, it is going to inhibit a large group of proteins. And this inhibition of the synthesis of the large group of protein will lead to the bacterial cell death. Yeah, in most of the population. But, and here is the catch. There is an increment in the synthesis of a small group of the protein in a smaller population in a given consortium, which can lead to the death proteins, or it can lead to the survival proteins. Now, these death proteins and survival proteins are differentially regulated. Now, too many questions will come to your mind. How this bacteria is deciding whether I should give rise to survival proteins or death proteins? Now, this is extremely complicated in this particular forum. For this, you need to understand the concept of bacterial aging. You know, in a given community, like the human population age, you have great-great-grandfather, grandfather, mother, father, children, like that. In a bacterial population, there is a similar hierarchy. So you have very old population, then you have a population which is less older, juvenile. It is difficult for you to understand because you will ask a question, a bacterium dividing into two, where is the mother bacterium? There is no concept of preservation of that mother figure. One bacterium becomes two daughter. Okay, so it's very difficult. Again, as time, if sometime I get a chance, I will explain you what is meant by bacterial aging. So there are phenomena of bacterial aging, and it looks like that what I speculate, we are ourselves doing certain experiment to show that the young bacterial population in a given cohort or a consortium is expressing these survival proteins. And the death proteins, which are expressed in much older population, then they're dying, they are releasing the nutrient for the nourishment of the younger population, a kind of an altruistic, manner that population is saving the real young ones. And that is how you know that at any given point in time till this earth exists, can we ever get rid of bacterial diseases. And you also know that why the bacteria can never become extinct, because you always have this survival population. Now, this survival population, this is like rising from ashes. You have read Harry Potter. Many of you are fond of Harry Potter. And you know that rising from ashes, right? The bird, phoenix, it rose from ashes. It's just like rising from ashes. You will think, and we are fooled that we take the antibiotic and we say, oh, bacteria dead. Where is it dead? Again, it will relapse and again it will come out. So that circle will never go. And this small population will ever persist, and then will become resistors, and then will become superbugs. Can you ever get rid of this? As if you ask me, after my own association with bacteria for such a long time, I will give a very simple, plain answer. No, there can be $1 billion spent on this. So that is different other alternatives. This warfare. This small population is always there. Yeah.
Ma'am,
Yes,
your
yes.
voice is not audible, ma'am. Some network issues.
Oh, when is it not audible? From
Just
this or
now,
from
just
before?
now, just now, ma'am.
Okay. Just now, right? Okay.
Yes.
So what I'm trying to tell is that if you ask me that whether there will be any end to this warfare of MDRs or XDRs, my answer is no. You can never have. Because this is like rising from ashes. You know, you all have read Harry Potter. Many of you are interested in this Harry Potter series. This bird, you know, rising from ashes, phoenix. It's just like rising from ashes. You will think that, oh, we have killed all the bacteria. See, bulk is dying. So what? We are safe. You're not safe. Nobody is safe. The reason is this small population exists. And it is this small population which is giving rise to superbugs, right? So where is the end now? Where is the end? One can spend $1 billion and there will be no end to this. And that is the only reason why now we have to be a little bit more intelligent and lose our supreme being that we are, and we can do everything. Just come to the ground level and say, no, we are not. Even to tackle this, we are not. Then for what? It's very difficult. So that is why we have to come to a harmony. Now, okay, I hope that this is clear to you, that the majority of the population dies, but there is a small population which survives because of the upregulation of the survival genes. And also the dead populations contribute to the simple nutrient. You know, bacteria are very simple. Simple beings are very elegant, by the way. So the simple nutrients which they release, the small population, they feed on these nutrients and they beautifully survive, right? Now, what is it to do with quorum or to with the density? This question will, this experiment will tell you. I mentioned to you, anything that you want to relate to the quorum, you have to relate it with the density. It should be density dependent phenomena. Now, the experiment which was put up is extremely simple here. You take a different density of bacteria. Don't concentrate on this mutant now, Mazef. Only look at the wild type E. coli bacteria, okay? And in the x-axis is a different density, 10 raised to 8, 7, 6, 5, and 4. And y-axis is a CFU, colony forming unit, where you are calculating the percentage of survivors. Again, by simple plating experiment, entire population is with a different density of bacteria is subjected to antibiotic, all the antibiotics, trimethoprim, nalidixic acid, then penicillin, streptomycin, whatever you can name it, it is there. Now, if you really look at the percentage of survivors, look at the 100% survival when the antibiotic is there, but the bacterial number is only 3 into 10 raised to 4, that is 30,000. Even the bacteria is not responding to the antibiotic when it is 3 lakhs, 3 into 10 raised to 5. They start responding to antibiotic in number dependent or the density dependent when it is 3 into 10 raised to 6 onward, and then the death happens when it is at a high density or at an optimal density of 3 into 10 raised to 8. Okay, this you keep it in mind. What does it mean? This means extrapolate it to our own situation when we are sick. Do the doctor know what is the density of the bacteria in the blood? Do they know what number we have? Do they know what is the number or the density in the organ? No, there is no way to know, but we get antibiotic. So you know now why there is no action. There will be no death. Only when the bacteria reaches such a density, there is a death. But if you make a knockout in this massive system, the toxin antitoxin system, which senses this entire stress and leads to a population dependent death, there is no death. What does it mean again? That means that this massive system is a population dependent system which induces a death in a given population by a molecule. Because any quorum system should be dependent on a molecule which is secreted. That is for sure, yeah? It is like a public good which you share. Quorum cannot be kept with yourself. Quorum has to be distributed so the entire community benefits. So what is this that this massive system is making that is leading to the death in a population in this particular concentration? What is it? And that's what made it a huge. Science is a very reputed journal, right? So to answer this question, what they did a very simple experiment. They took a dense culture. When I mean dense means 3 into 10 raised to 8. Okay. And you see that really in the dense culture, with respect to, in the presence of antibiotic like chloramphenicol or trimethoprim, they die. When they take a culture which is not dense, that is 3 into 10 raised to 4, you call it as a sparse culture or light culture, dilute culture. And they don't die. They don't respond to the antibiotic at all. When you take the supernatant, what is the supernatant? Supernatant is the spent medium. Say, for example, you are taking a LB tube with 10 ml of LB, then you are growing your wild type and you want to collect the supernatant, you centrifuge the tube and you take that clear fluid. That is called as the supernatant. Supernatant research is very important. It gives you a lot of insight. So when you take a supernatant from this dense culture and then you put it in this sparse culture, that is 3 into 10 raised to 4, the bacteria dies. That means what? That means that there is something produced in this dense supernatant which is being sensed by the bacteria so that the population is dying. Now, this dense supernatant containing molecule is a matter of research now. What is it that it is making? That now if you put that dense supernatant into the light culture or the dilute culture, the bacteria dies. Okay, and this is not being sensed if you make a mutation in the massive. In this particular mutation, even the molecule is not made, it is not sensed, nothing happens. And they all beautifully survive at a dense culture, light culture, every culture in the presence of all kind of antibiotics. Question is, and this is a kind of a very important, finding, what is this molecule? Now, that is what I mentioned to you, that the supernatant research is very important. They realized that this molecule, which they termed it as a EDF or extracellular death factor, if in this y-axis you see that the EDF is produced. So the bars are the production of the EDF. It is a unit per ml. And this particular box is the diamond. These are the growth of the bacteria. Okay, so this is OD600, growth of the bacteria at a different time in minutes. And you have the EDF production. You see that the maximum EDF is produced when the bacteria is in the logarithmic phase. Yeah, and then the production of this toxin goes down. So this is also one of the very interesting facts, that you do not see a rapid death when the bacteria achieves the stationary phase. So the stationary phase bacteria, they just stay stationary. They do not undergo this PCD. They are resistant to the PCD because they do not have this EDF activity. And that is also one of the reasons that why many of the antibiotics are added when the bacteria are logarithmically growing. and not when they are in the stationary phase. Many of the antibiotics don't act. Now, the bulk of the population that is dying is because of the production of the EDF. Okay, so this is there. But now the question is, what is this molecule? Now let us look what is this molecule. They are very lucky to figure out that what this molecule is. The scientists were very lucky. When they took the supernatant and do a hell lot of data they generated, did the mass spec, they figured out that EDF is nothing but a linear peptide. It is a pentapeptide. It is aspargine, aspargine, tryptophan, aspargine, aspargine. That's it. It's a linear pentapeptide. It is such a short peptide. Of course, that's what somebody asked. No, what about the molecules of quorum sensing? Is it only acylated homoserine lactone? What happens in the gram positives? In fact, these quorum sensing molecule can be anything, just not the AHL. Like in this paper, they figured out that this molecule is a simple linear pentapeptide. This is the EDF, which is secreted, extracellular death factor. It is secreted by this massive system. And when it is secreted, it is sensed by the bacteria to induce the population-dependent death. Now that you have these very beautiful molecule, it is a small linear peptide, one can synthesize in the lab. Any peptide synthesis lab, any simple lab will synthesize and give you this molecule because it's just a 5-peptide. And it's very simple chemistry of peptide synthesis. So the authors did the same. They brought this peptide and in bulk, they made it in bulk. And then you see here, now work with supernatant, because supernatant is sometimes difficult, you know. You have many other substances there, but if you have a purified chemical, you can go away with the supernatant. Dense culture, 3 into 10⁸, the bacteria dies in the presence of antibiotic. But again, look here, not all, by the way. So that surviving population is the key here, which you can never get rid of. Now, I also don't know how do you tackle or design a strategy to get rid of that. Diluted culture never responds to antibiotic, they never die. But if you add this EDF in the diluted culture, they die. That means the EDF or the extracellular death factor is the signal for the death of the bacteria. And the moment you knock out this massive, they become non-responsive even to the extracellular EDF. The entire cascade of the massive system governs the process of not only producing the EDF and secreting, but also sensing the EDF by the other group of bacteria, and also giving that rise to the small survival population. So it is doing all of that it is doing. Okay. So what I'll do is I'll again stop here. And now I have made you understand that what is the reason that we cannot get rid of the bacteria. This you have looked by yourself, that a dense culture responds to the antibiotic, but not the diluted culture. That means whatever we have learned about the traditional role and the effect of antibiotic is absolutely not correct. And you also will correlate it to our existing problem, what we face of not able to get rid of the bacterial infections and not able to get rid of the AMRs and the XDRs and the MDRs, because the superbugs are generating from this population only. Now, what is the strategy that one can take? That we need to understand to figure out that it is indeed end of the disease. And my answer is that you can generate any new generation drug. Maximum within 3 months, you will get a resistance against it, because it's a right to live and they are going to do everything to make it happen. How do you get rid of this and this so-called superbug small population? That is a matter which I think only, some really enlightened thinking will tell us. But is it really approach is right that we are doing to have this constant warfare is my question. And I think that the approach itself is not right, and that is the reason that we are not able to get anything done. So I will stop here and now I will take questions. So I hope that I was able to convince you about the programs happens in the real population that is dependent on the density. And you also know now that how difficult it is even to treat anything, and how any disease without knowing the bacterial numbers and simply gulping antibiotic or giving intravenous injections, how we have contributed ourselves and created our own niche and both the topic of quorum and the kind of a novel tackling approach, so that we are at least in harmony and we do not suffer so much when it comes to the bacterial diseases. The same will be true for even viral diseases, and the same will be true for even parasite, but because they have vector, they are bigger, they are still a little bit easy to tackle, but not bacterial disease. Okay, so thank you very much. Now I will stop sharing and I will be opening to questions. So colleagues, kindly, okay, great. So we have questions here again. Dr. Abhilash, I'll read the question. In my limited knowledge, it is safe to assume that bacteria is a supra evolutionary being. Super evolutionary, it is true that it has a rightful place in our ecosystem to help us grow. Extremely important. See, Dr. Abhilash, if I today get rid of my gut flora, I am dead. Every single individual, be it ant or be it honeybee or be it mosquito, have their own gut flora. And that flora is keeping us alive. You know why this Yakult company is making $1 million by advertising? Take this probiotic, you will be, that is rubbish. But still, because that is a live bacteria which you are drinking. We are what we are is only because of them. So they are helping us to grow in a tremendous way, be it a plant or be it anything. Now there is a huge research of gut microbiome, right? Malarial parasite, which is harbored in a mosquito, they contain a bacteria. It's called the Vauchereria bancrofti or whatever, they contain a bacteria. Now the bacteria is important for the malarial parasite to stall their growth. Actually, when they get rid of the bacteria, the malarial parasite, they become hyper. Similarly in ant and similarly in honeybee, similarly in Drosophila. So yes, very rightfully, they help us to grow and they help us to have an ecosystem. And now, so okay, so I would further say that programmed cell death is, yes, very true. It is not some kind of a shabby death process. See, death also comes with a dignity. It is a decision, and the decision has nothing to do with their so-called thought action, like inhibition of transpeptidation, cross-linking, or binding to the gyrase, or doing this or doing that. No, it is a decision which is very consciously taken, and that community decision is taken by the bacteria to give rise to a small population, a young population, which will survive further and will take the life further. So it is, everything is programmed, nothing is shabby. Same is in case of in bacteria, that it is an extremely programmed cell death. Yeah. I was hoping more questions in this.
Any queries? Audience?
Yeah. There is one more question. Yeah, keep writing, colleagues. Don't hesitate. Yeah. So Survachala, thanks for asking. What are the conditions or reasons that lead to programmed cell death in bacteria? A good question. One condition is your antibiotic stress. Now they have figured out that any antibiotic is a major stress because we are struggling with it. We are struggling with our AMRs. We are struggling with infectious disease, but we are not understanding the fact that the bacteria we live with in ourselves is actually robust to make us what we are. And we should not destroy our gut flora. Colleagues, one take-home lesson: never ever abuse your gut flora. Don't do that. High-fat diet, abuse of alcohol, everything will lead to the perturbation of the flora, and you will slowly see the change in the lifestyle is leading to the change in your mood, change in the performance and everything. So be very careful with and preserve your gut flora. This is your second organ, by the way. It's given a status of a second organ in the body. So brain is one and this is the second. With this, brain cannot work. So there's a beautiful gut-brain axis. Okay, now coming to your question, what can give rise to programmed cell death? It is the stress which is studied the most is the antibiotic stress, but it is any kind of stress. Now tomorrow it can be heavy metal toxicity stress, it can be a pH stress, or it can be a high acid stress. So any kind of stress gives rise to these kind of the programmed cell death in bacteria. Yeah. And you can use different examples. So in this case is MASI-MASF toxin antitoxin systems. Like that, there are many modules of toxin antitoxin in many bacteria. And one can in fact take this system and have newer system in place to understand the programmed cell death.
Any questions?
Okay.
Audience?
No more questions. Everybody is tired. We are sitting from 10 onwards. Okay. Yeah, I don't see any more questions.
Okay, shall we wind up, ma'am?
Sure. So if indeed, if there are no more questions, so colleagues, kindly the take-home lesson. Okay, one more. Coming up. Great. Apoptosis, autophagic cells, death, and regulated necrosis, the differences. All these processes that you have mentioned, the apoptosis is in the mammalian system, and you know that there are a lot of programmed genes which are induced in the cell. Autophagic cell death is a process where the cell is autophagic, means taking its own molecules which are not good for the cell, giving it an autophagic machinery and clearing it off. That is an autophagic. Autophagy also comes in various colors. Mitophagy, xenophagy, perturbed mitochondria. Bad mitochondria are cleared by mitophagy. Xenophagy is exogenous material. Autophagy is the damaged organelles of your own system, own cell. They are going clearance by the autophagic machinery. Autophagy is to clear the cargoes which are faulty. Now, regulated necrosis, where apoptosis, the major difference between the apoptosis and the necrosis is in apoptosis, there is no release of the content. It is always like a condensation. Everything is within and nothing is released. Whereas in necrosis, it is every content comes out. So the cell will burst and it will just come out. And all the three processes are pertained to mammalian system. But that is what the programmed cell death is, the word which is given to the bacteria. And you know that in the programmed cell death, there is actually a release because they release the nutrients so that the surviving population can act on. So, ma'am, is programming cell death occur in cancer cells? Yeah, there is a role of the programmed cell death in cancer, because in cancer what happens, they bypass the PCD, mechanism. So there is an induction of the programmed cell death. That is one of the therapy that they use, that you can induce the programmed cell death or apoptosis in the cancer cells to curtail or to control the growth of the cancer. So very much, lot of the drugs, they aim towards inducing the apoptosis in the system. After reading Emperors of Malady, yeah, Siddharth's book, my favorite book too. This is the single most engaging interaction. Oh, thank you very much, Dr. Vilas. Emperors of Malady is one of my very favorite book, and he himself being a doctor. And, you know, I really hope that the doctors have that kind of empathy and that kind of sense to understand the meaning of science and the meaning of life. Yeah, thank you very much, Dr. Vilas. It is one of my favorite books too. Yeah, thank you so much. Which cells cannot be killed by the programmed cell death? Which cells cannot be killed? Now, all cells can be killed if you induce the PCD. The PCD resistant cells are the one where they have a mutation in the genes which is responsible for the PCT. Like say, for example, in the mammalian system, there are a lot of these genes, Bax genes, the caspase genes, which help in the cells to undergo programmed cell death. If you have a mutant in a multiple pathways, then they become resistant to PCT. And that is how the cancer cells become resistant. See, we have not only done damage to the bacteria, we have done damage to this cancer therapy also, because these cells are becoming now, cancer cells are becoming resistant to the drug. And again, our own cells becoming hostile to us due to whatever reason. This is a bit, philosophical question to understand the right to live for everybody. And then who suffers in this context? Is there really a word called suffering? Suppose a human suffers, so are mosquitoes, so are ants, and so are bacteria. Now, who's suffering? Just because we are here and we can make our presence and we have a loud voice, are we supreme? And the answer is actually no. The moment this can be understood, everything will come to that level. But I hope that this will. So yes, so answer to the question is, all the mutants which cannot respond to them, they are resistant to the PCT, but then you have a counteractive other genes which can somehow then again make them sensitive to the PCT. Yeah, cancer cells. You wanted to know what happened in cancer cells, Pushpa? In cancer cells, many drugs, they induce the programmed cell death so that they can undergo death. But in cancer cells, it is just not the cancer cells. The cancer stem cells are the one which gives rise to the new progeny or the new cancer. Now, it is just not the tumor. The tumor microenvironment relishes, the stem cells relishes that niche. And it is the stem cells from which everything sprouts. How do you take care of the stem cells now? That is the question. Yeah. How are you going to? So, yeah, you can use combinatorial therapy to, in fact, get rid of things and just pray, saying that nothing should pop up again, because it is again no exogenous material. It is our own cell becoming hostile. So what is yours? Question is, what is yours? What you call as yours, the body as a yours has already turned hostile. Then what is yours? So better to remove everything with this belongingness, not even your own self. Yeah. Thank you so much.
Thank you, ma'am. Thank you so much, ma'am, for enlightening us and for your excellent presentation and nice explanation about quorum sensing and the program cell death in an excellent way. It was really a mind-blowing session, ma'am. So we learned more and more from your presentation, ma'am. On behalf of the Biochemistry Department and the Sacred College, once again, I extend my sincere thanks to you, ma'am.
Thank you very much. My heartfelt thanks to all my colleagues who participated, and especially Dr. Abhilash, because you are the only one, I think, from humanities, and you brought in a different perspective, and that perspective now needs to be brought it in hardcore science so that we can think different. And to all my other colleagues who have asked wonderful questions, and it was an amazing, engaging discussion. So thank you so much. Thank you, Dr. Gomti. Thank you, Dr. Niaz, for this invitation, and I indeed enjoyed. Thank you.
Thank you, ma'am. Thank you. So
thank you very much. See you. If you need anything, kindly write me an email. If you need any manuscripts, papers, I will communicate with you. Yeah. Thank you so much.
Thank you, ma'am.
Bye-bye. Take care. Take care. Bye.
So this is an...
Thank you. Thanks for joining with us, ma'am, even your busy schedule.
Thank you very much.
She is on the board towards Chennai. Ma'am is on the board towards Chennai. Happy Bon Voyage, ma'am. Welcome to Tamil Nadu. We congratulate and all the very best for your DHT meeting. Thank you for joining with us, ma'am.
Thank you so much. Bye-bye.
Yes, sir.
Take care. Bye.
Dear audience, this is a time for lunch break from 1 up to 2 p.m. Next section will begin sharply at 2 p.m. So kindly enter through the same link. So tomorrow evening only we will post the feedback link through chat box. Thank you.
Thank you, ma'am. Thanks, ma'am. We can meet at 2 o'clock. Thank you. Thank you.